Bom, já temos 110 pessoas dentro da plataforma. Meu nome é Aline, eu sou coordenadora da Academia Finatec, responsável pelo evento, e gostaria de dar algumas orientações a vocês que estão conosco aqui na plataforma. Antes de mais nada, em nome da Academia Finatec, em nome da comissão organizadora do seminário, sejam muito bem-vindos. Nós agradecemos a participação de vocês, a intenção de estar no seminário. Eu gostaria de dar algumas orientações. O nosso seminário ele é traduzido do início ao fim, na língua português e inglês. Nós temos aqui embaixo, na barra do Zoom, uma opção de escolher a língua a qual você quer acompanhar o evento. Então, tem um botão de tradução embaixo, onde você vai clicar inglês ou português, e também decidir se você quer ouvir o som original da língua original. O que isso quer dizer, Aline? Quer dizer que, se eu escolher o português, que é o meu caso, eu posso ouvir o som do palestrante em inglês no fundo da minha gravação, da minha palestra. Então, você pode ou não desativar esse botão. Como falado anteriormente aqui no chat, a nossa comunicação será através do bate-papo. Também na barra inferior tem um ícone bate-papo, onde você pode falar tanto com os seus colegas como um todo, escolhendo todos, ou particularmente com alguém através do chat, onde não é acompanhado pelos demais participantes. O nosso mediador e o nosso professor responsável pelo evento hoje é o professor Ricardo Dias. Eu quero te dizer que você pode ficar à vontade para participar com perguntas, com considerações, através do chat. Você pode ficar com a sua câmera ligada, mas o seu microfone estará desligado, justamente por conta das participações via chat. Então, eu desejo a vocês um ótimo evento, um dia próspero, um dia produtivo, e sejam muito bem-vindos. Professor Ricardo, bom dia.
Bom dia a todos. Eu também gostaria de dar as boas-vindas a todos os participantes, aos palestrantes e em nome da comissão organizadora. O nome do nosso webinar é Advances in Surveillance and Control of Vampire Bat Rabies, que acontece no dia de hoje, e Insights into the Economics of One Health Surveillance Programs, que ocorre no dia 30, na sexta-feira. Então, esse evento, ele está no âmbito de um projeto financiado pelo Ministério da Agricultura, Pecuária e Abastecimento, em colaboração com o IICA, e é organizado pela Finatec, a qual eu agradeço muitíssimo pelo profissionalismo, pela ajuda, pelo programa de pós-graduação em Saúde Animal da Universidade de Brasília e pelo programa de pós-graduação em Epidemiologia Experimental aplicada às zoonoses da Universidade de São Paulo. Bom, meu nome é Ricardo Dias, como eu falei antes, eu sou docente dessa universidade, da Universidade de São Paulo. Então, no primeiro dia, nós focaremos mais em vigilância de raiva em morcegos hematófagos. Nós teremos 3 palestras e 4 palestrantes. A última palestra vai ser dividida, né, o tempo. Eu só reitero que a primeira palestra vai ser falada em português, para aqueles que estão ouvindo no YouTube a transmissão, e o restante das palestras, inclusive na sexta-feira, serão faladas em inglês. Então, as perguntas no chat podem ser escritas em português ou em inglês. Como a Aline mencionou, nós temos muitos participantes, então essas perguntas serão selecionadas. Infelizmente, talvez nós não consigamos fazer todas, mas nós vamos fazer um esforço para fazer todas essas perguntas, se possível. Antes de apresentar o primeiro palestrante, Felipe Rocha, eu gostaria de fazer uma apresentação né, de um currículo rápido, né, de cada um dos palestrantes. O Felipe Rocha, ele é consultor de medicina veterinária e de zoonoses no Centro Pan-Americano de Febre Aftosa e Saúde Pública Veterinária, o Panaftosa, que faz parte da Organização Pan-Americana de Saúde, a OPAS. Felipe completou a sua graduação na Universidade de São Paulo em 2012, em medicina veterinária, e o seu mestrado em epidemiologia veterinária em 2018, sob a minha orientação, também na Universidade de São Paulo, com o tema de biologia dos morcegos hematófagos. Então, eu passo a palavra ao nosso primeiro palestrante. Felipe, bom dia. Gostaria de te dar as boas-vindas.
Olá, professor.
Tudo bem? Obrigado pela sua participação, Felipe.
Agradeço o convite, agradeço também a organização do evento, ao professor Vitor e a você, professor Ricardo, por me convidarem a participar desse evento. Para a gente do Panaftosa é de suma importância estar nesse contato direto com a academia e também com os programas, então agradeço bastante o convite para considerar o nosso centro para a participação desse seminário. Vou, então, compartilhar a tela do meu computador para dar início, então, à palestra. Gostaria também de agradecer aos participantes desse evento por disponibilizarem, né, o seu tempo para ouvir a gente. Então, meu nome é Felipe Rocha, como o professor Ricardo apontou. Vou desligar a câmera momentaneamente para fazer a apresentação, depois eu volto a abrir. E eu vou dar uma palestra sobre a perspectiva regional sobre a raiva transmitida por morcegos hematófagos, em especial desmodos rotundos, aos animais de produção suscetíveis nas Américas. Então, a Organização Pan-Americana da Saúde, ela existe há 108 anos, ela é anterior à própria Organização Mundial da Saúde, e ela é a representante, o escritório para as Américas da Organização Mundial da Saúde, né, e ela atende aí 48 países e territórios que são membros da organização, quase 1 bilhão de pessoas, né, tem que completar aí 118 anos de história. Especificamente sobre o Panaftosa, né, ele foi criado em 51, inicialmente no combate à febre aftosa nas Américas, e nos anos recentes, a partir de 98, mais ou menos 20 anos atrás, passou a incorporar também a área de inocuidade dos alimentos e a área de zoonoses, que é onde eu trabalho especificamente, né? E ela fica localizada, a Panaftosa, ele fica localizado em Duque de Caxias, na Baixada Fluminense. Então, a raiva ela é uma enfermidade aí com múltiplos ciclos e várias faces, com vários animais envolvidos aí no processo de transmissão e também de spillover a outros animais, mas a gente vai falar especificamente aqui do ciclo que ocorre no meio rural, e quando eu digo meio rural, eu estou falando do meio onde há a produção de animais domésticos, né? E a gente vai deixar de lado aqui os ciclos que compreendem o meio urbano e o meio silvestre de mata fechada, né? Então, desmodos rotundos, que é o principal transmissor da raiva, o principal ator envolvido nesse ciclo epidemiológico da raiva, ele é um animal que está presente em 22 países das Américas e é um animal que se adaptou às alterações ecológicas que o homem promoveu nas Américas desde da sua chegada em massa aqui, né, nos últimos séculos, especialmente com a expansão da produção animal no nosso continente. Ele se adaptou a essas alterações, também alterações climáticas, e passou aí a se tornar um animal muito frequente no nosso território. Então, a situação epidemiológica regional, durante 2015 a 2016, foram reportados, lá que a América Latina e Caribe, foram notificados mais de 7 mil focos unicamente em 2.900 focos em animais de produção, sendo que mais de 7.000 animais foram testados para possibilidades de outros focos, né? Então, aí você já vê uma diferença entre a quantidade que foi positiva, a quantidade que foi negativa, que foram outras causas neurológicas em animais, né? A gente também observa uma subnotificação muito grande de casos nesses 22 países das Américas, e se a gente pegar só o Brasil, um dado do IBGE, de uma média aí de mais ou menos 3 trabalhadores rurais por prédio, por unidade produtiva no Brasil, a gente leva em consideração a quantidade de focos que foram reportados no Brasil, a gente tem aí uma estimativa da quantidade de pessoas que foram expostas ao risco da raiva, né, em contato direto com esses animais domésticos. E um ponto importante aqui é que o Brasil se destaca na quantidade de focos notificados nesse período. Entretanto, quando a gente relativiza isso pela população e a gente olha a incidência de casos por animal, o Brasil, por ter esse território continental e uma população muito grande, né, de animais domésticos de produção, ele já não fica em destaque. E a gente vê já países como México, Belize, Guatemala, Panamá e Peru, que passam a ter aí uma relevância maior na questão da incidência animal, né? E os impactos que são sentidos pelos produtores e pelos países no tema de raiva, a gente vê que são aproximadamente 500 milhões de herbívoros domésticos que habitam essas áreas de risco, né, para raiva transmitida pelo morcego hematófago, com uma mortalidade anual de mais de 100 mil animais aí por ano, estimadamente, e em valores atuais a gente está falando de uma perda aí de quase 100 milhões de dólares, unicamente com a morte de animais na América Latina. E como que os países respondem a esse problema, né, a essa questão? Então, a gente pegou e fez uma busca nesses 22 países com desmodos rotundos para verificar o que a legislação de cada um desses países dizem sobre o enfrentamento à raiva transmitida pelo desmodos rotundos. E dos 22 países, em 10 países você não encontra referência de legislação especificamente sobre a raiva de herbívoros. 12 países você encontra legislação, só que desses 12, apenas 7 países têm programas estabelecidos com as ações e com as atividades propostas no combate à enfermidade. Desses 12 países com legislação, existem 5 pontos em comum entre eles. Primeiro é que a raiva é uma enfermidade de notificação obrigatória de casos suspeitos. Segundo é que não são todos os 12 que apresentam essas 5 características, tá? São características em comum em uma maioria desses 12, né? Campanhas de vacinação em áreas com histórico de raiva, a realização de ação de quarentena dos prédios com focos, ênfase em boa parte nas ações de atenção aos focos, e também estabelecimento de ações de controle da população de desmodos rotundos. Agora, um pouco sobre cada um desses 5 temas, desses 5 pontos, e a nossa visão regional sobre esses 5 detalhamentos. Primeiro, a quarentena de prédios. A gente entende que o herbívoro doméstico ele é a última etapa de uma cadeia de transmissão, então não ocorre aí uma transmissão, a não ser em casos muito específicos, em situações muito especiais, de bovino para bovino, ou de animal doméstico para animal doméstico. Então, quando você isola um prédio, pensando em conter a infecção, você não vai impedir a proliferação dela por meio do isolamento daquela unidade produtiva, e você pode estimular ainda a subnotificação de casos, né? E um argumento muito utilizado são os riscos relacionados à saúde pública pela movimentação desses animais. Mas a gente entende que uma boa ação de integração com saúde pública, né, na atenção da pós-exposição, mitigaria o risco aí à saúde pública. Outro ponto interessante sobre a questão da prevenção e a atenção do foco, né? Então, como a gente tem programas e legislações que dão uma direcionada e uma ênfase maior à atenção de foco, a gente sente, os países dizem para a gente, os chefes de programa, muitas vezes dizem que trabalham como bombeiros, indo de fogo em fogo, incêndio em incêndio, como se estivessem enxugando o gelo, né? E a prevenção, muitas vezes, ela é deixada um pouco de lado, e uma ênfase maior é dada à atenção de focos, isso cria um desequilíbrio na atenção à enfermidade. Sobre o controle populacional de morcegos hematófagos, o que a gente vê é que existe uma dificuldade para a manutenção dessa ação por parte dos países, seja porque ele é realizado de maneira pouco inteligente, não sistemática, e também isso acaba gerando aí a visão errônea de que a ação ela é muito onerosa e pouco efetiva, quando, na verdade, ela é a única ferramenta que a gente dispõe para controlar uma população de animal silvestre que cresce artificialmente e traz aí ameaças, problemas à sanidade animal e à saúde pública. Experiências do passado mostram para a gente que, quando bem implementada, de forma inteligente, sistemática, ela pode, sim, trazer benefícios grandes no controle da enfermidade. Um pouco sobre o diagnóstico que é utilizado pelos países das Américas, a gente vê aí nesse gráfico, o lado amarelo, desses meios círculos, eles indicam aí a proporção de países que utilizam essa ou aquela técnica. E a gente vê que, para a área da defesa animal, de sanidade animal, a imunofluorescência e o isolamento em camundongo são os dois métodos mais empregados na América Latina para diagnóstico da raiva. Então, a nível de país, dos países das Américas, o que a gente vê? Os países eles têm entendimentos diferentes sobre o problema. A gente vê uma heterogeneidade de práticas e metodologias aplicadas ao enfrentamento da raiva e dificuldades variadas, múltiplas, enfrentadas pelos países, inclusive algumas que são comuns. Então, isso dá à América um problema único no mundo, que é a raiva transmitida por desmodos rotundos, né? Então, regionalmente, como que a OPAS, por meio do Centro Pan-Americano, né, a sua divisão de saúde pública veterinária, como que a gente poderia apoiar os países nesse tema? Então, no dia 23 de julho do ano passado, nós realizamos uma reunião virtual com os diretores dos programas de raiva, tanto de saúde quanto de agricultura, para falar especificamente sobre esse tema. A gente fez já 5 reuniões do ano passado para essa, todas em caráter virtual, pela situação sanitária que a gente vive. A segunda, no caso, foi sobre o tema específico de raiva de herbívoros. E dessa reunião, os diretores dos programas de raiva deram a recomendação de que o Panaftosa, então, organizasse, em conjunto com países, o desenvolvimento de um programa regional de combate da raiva em herbívoros, né? E os principais objetivos desse programa seria apoiar os países no desenvolvimento e na atualização de programas nacionais, lembrando daquela informação sobre a informação de legislação e de programa dos 22 países das Américas, e também o desenvolvimento do programa regional seria para realizar fortalecimento político dos programas nacionais, né, dando respaldo a eles para estabelecimento de suas ações, harmonizar as estratégias entre os países, promover o intercâmbio de informação entre eles e o monitoramento do progresso que cada país vai tendo no tema. Então, como que é o processo de construção do programa regional? Então, no dia 23, como eu falei, foi a segunda reunião, que saiu a recomendação que o Panaftosa formasse um grupo de trabalho para a elaboração do programa regional. Durante o segundo semestre do ano passado, e aí a gente coloca aqui os países, os 8 países que compuseram o grupo de trabalho, todos eles foram países que se voluntariaram a participar. O Brasil foi um deles, aí na representação da doutora Ellen, que foi representante por parte da agricultura do Brasil e participou da construção dessa proposta de programa regional que eu vim apresentar para vocês, né? Fora o Brasil, a gente também teve a participação da Argentina, Colômbia, Equador, México, Paraguai, Peru e Uruguai. E a gente realizou 6 reuniões para a construção dessa proposta de programa regional, né? Ainda durante esse semestre, é o momento atual que nós estamos, a gente está fazendo uma análise para a versão final do documento do programa regional, e depois a gente vai apresentar isso aos demais diretores de programas de raiva por parte do setor de agricultura, para consideração deles, e, por fim, a aprovação do programa regional numa sessão virtual Redipra, que é a reunião dos diretores dos programas de raiva das Américas. E, a partir da aprovação do programa regional, a gente passa a implementar as atividades e fazer o segmento dos países de acordo com o previsto no plano. Então, considerações para a construção do programa regional. A gente tem que lembrar também da questão da heterogeneidade e dos diferentes entendimentos sobre a raiva que existem nas Américas. Então, o primeiro ponto é uma visão macro-regional, né? Então, o programa ele tem que levar em consideração que ele deve englobar vários países e deve monitorar o avanço regional como um bloco, como uma unidade, né? A espelho do que a gente vê já nos programas de febre aftosa, por exemplo, que a gente tem o PEFA, que é o Programa Hemisférico de Erradicação da Febre Aftosa, e na parte da saúde, o controle da raiva canina, né? Que são dois programas que o Panaftosa tem atuação direta e que tiveram grande sucesso na nossa região. Levar em consideração também que os países estão em níveis diferentes em programa. Como eu falei lá atrás, você tem países como o Brasil, com programas já bem estabelecidos, bem fundamentados, e com longa aplicação ao longo dos anos aí desse programa, mas você tem países que não têm nem menção à raiva em sua legislação. Então, o programa ele tem que tentar englobar esses diferentes caráteres aí em questão de legislação dos países. Levar em consideração também que a gente está falando de um continente com características ecológicas e epidemiológicas distintas entre os países, e até dentro do próprio país, a exemplo também do Brasil, por toda a sua dimensão, por todo o seu território continental, você vai ter diferenças ecológicas e epidemiológicas dentro dos países. Por fim, a estrutura dos serviços, né? Cada país tem uma estrutura aí a nível ministerial e de execução das ações de defesa animal que funcionam de maneiras distintas. Então, a gente iniciou o trabalho imaginando qual seria o fluxo da ação de um programa. Então, a gente tem na parte superior aí, em amarelo, a parte de vigilância epidemiológica. Então, você teria aí a vigilância das agressões, dos abrigos e desmodos rotundos e dos casos suspeitos de raiva. Lembrar que à esquerda a gente está falando da detecção de sinais antes da ocorrência de caso, e a partir do momento que você tem casos suspeitos, você já está falando de sinais após a ocorrência, então, dos casos, né? Quando a gente fala, então, de a vigilância de agressão e de abrigo de desmodos rotundos, a gente entende que a agressão é uma ferramenta para nos direcionar a identificação dos abrigos, e os abrigos também a nos direcionar aonde estão ocorrendo as agressões. E a partir da identificação desses dois pontos, você pode entrar com as ações de prevenção. As ações de prevenção, por sua vez, elas vão alimentar aí um banco de informação embaixo, em vermelho mais escuro, assim como vão direcionar a integração com saúde pública e educação sanitária, né? Agora, olhando à direita do nosso fluxograma, a partir de um ponto que você tem casos suspeitos, você tem a confirmação do caso por meio de diagnóstico laboratorial, você tem a atenção, então, do foco, e da atenção do foco você deriva aí para integração com saúde pública, a educação sanitária também, e a alimentação de sistema de informação. Tanto as ações de prevenção, como a atenção de foco, como esses sistemas de informação, eles direcionam, alimentam a própria vigilância epidemiológica, formando aí um ciclo, né, sustentável aí de geração de informação, e essa informação gerando ações direto no campo. Então, olhando aqui mais ou menos, a gente vê vigilância na parte superior, à esquerda a gente vê ações de prevenção, à direita a gente vê atenção de foco, diagnóstico, abaixo a gente vê aí o sistema de informação. A gente começa a idealizar aí, pensar como seria a estrutura de um programa regional. Então, a gente estruturou ele baseado em 6 pilares centrais. Então, o primeiro deles seria a legislação, o segundo seria a vigilância, o terceiro seria as ações de prevenção, o quarto, diagnóstico, o quinto, atenção de foco, e o sexto, sistemas de informação. E complementar a eles, né, transitando em todos eles, a gente tem o que chamamos de componentes transversais. Eles não formam pilares únicos de atenção, mas eles estão permeando, eles estão incluídos nesses 6 pilares, que é a capacitação profissional, a educação sanitária e a integração com saúde pública. Então, como avaliar a situação atual dos países e o avanço dos programas? Então, observando esses 6 pilares e a gente estabelecendo eles como objetivos, fortalecimento da desenvolvimento da legislação, fortalecimento da vigilância, fortalecimento das ações de prevenção, fortalecimento do diagnóstico, fortalecimento da atenção de foco e desenvolvimento de sistema de informação aplicável, a gente tem o que a gente chama de os pilares, que seriam os objetivos do programa regional. Os objetivos, por sua vez, eles seriam compostos por metas, ou seja, ao país realizar um conjunto de metas que englobam aquele que estão parte daquele objetivo, ele alcança o objetivo. E as metas, por sua vez, são compostas por atividades. Então, qual seria a sucessão? Ao executar e implementar atividades, o país, por sua vez, implementa metas, e um conjunto de metas implementadas e operantes formam e compõem um objetivo cumprido. Então, todo esse conjunto de atividades, metas e objetivos, ele gera indicadores para a autoavaliação do país e como medidas de avanço, que somados, então, vão representar a região, isso somando as informações de cada país. Então, como eu falei aqui nesse slide, você tem atividades, um conjunto de atividades compõe uma meta, e um conjunto de metas compõe um pilar. E como eu falei anteriormente, a gente tem 6 pilares. Eu não vou ficar aqui mostrando cada pilar e suas metas e suas atividades especificamente, porque senão eu ia usar todo o tempo só para descrevê-los. Então, eu trouxe um aqui como exemplo, que é o objetivo número 2, que é o de fortalecer a vigilância epidemiológica de agressões, abrigos e desmodos rotundos e casos suspeitos de raiva. Então, ele é composto por 3 metas, a meta 1, a meta 2 e a meta 3, sendo a meta 1, monitorar a ocorrência de agressões por morcego hematófago. E aí a gente tem as atividades dessa meta. Então, é dispor de um sistema para receber notificação de agressões, dispor de um cadastro confiável de prédios ou unidades produtivas, dispor de profissionais capacitados para identificação de mordeduras. Então, qual que é a lógica qual seria a lógica do programa regional? O país, ao cumprir essas atividades que eu acabei de citar, ele cumpre a meta. E quando ele cumpre todas as 3 metas desse eixo, né, desse objetivo do programa, ele cumpre, então, o objetivo maior de fortalecer a vigilância, né? Então, veja que a gente tem aí uma dicotomia, né? Ou você cumpre, ou você não cumpre. Você tem isso implementado, ou você não tem isso implementado na questão das atividades. Mas se a gente junta as atividades, a gente vai ter atividade que é executada, atividade que não é executada. Então, por sua vez, as metas, elas não podem ser também dicotomizadas, elas não podem ser só meta cumprida ou meta não cumprida. Então, por isso, as metas, elas foram classificadas em 3, como meta implementada e operante, meta parcialmente implementada e meta não implementada. Então, eu trouxe o exemplo aqui de um país fictício para a gente observar aí como seria dentro dessa classificação de metas e atividades, como que o país estaria se classificaria. Então, ele não possui legislação para o tema, que compreende todas as atividades propostas pelo programa regional, apenas parte delas, mas acredito que poderá ter uma norma técnica completa aí nos próximos 2 anos. Então, aí a gente tem uma legislação que é parcialmente implementada, né? Ele só compreende parte das atividades propostas. Em questão de vigilância, que é a lupazinha ali, a gente vê que apenas a vigilância de focos é realizada, porque não tem na legislação uma especificação sobre abrigos de desmodos rotundos, nem de agressão, apenas a vigilância de casos suspeitos, né? Então, quando a legislação for alterada, ele vai ter que passar a executar a vigilância dessas agressões no monitoramento de abrigos, uma vez que a norma é implementada. Em questão de diagnóstico, nesse país fictício, não há execução de diagnósticos diferenciais, não realiza interlaboratórios para estabelecer os níveis de qualidade, de padrão dos seus laboratórios. Toda a demanda de diagnóstico é centralizada em um único laboratório central, nacional, que assim mesmo atende a quantidade de amostras a processar, uma vez que é um país pequeno. Então, ele realiza o diagnóstico, ele tem como realizar o diagnóstico, mas ele não realiza uma série de outras metas que compõem esse objetivo do diagnóstico. Quanto à prevenção, que é o ícone vermelho do escudo, e a atenção de foco, que é o extintor verde, ele, quando ocorre foco, o serviço oficial ele assiste aos produtores na aplicação de vacinas, realização de estabelecimento de área de foco, área de perifoco. Então, ele só tem aí cumprido a atenção de foco. E, por fim, ele não conta com um sistema de informação para coleta de dados e análise de informação. Então, essa é a descrição do cenário que a gente identifica nos países fictícios, e a questão da classificação entre implementada e operante, parcialmente implementada e não implementada. Então, a partir disso, a gente chega no grande produto do programa regional, que é uma matriz de autoavaliação desse país de exemplo. Então, olhando primeiro aqui para essa tabela de progressão à direita, você tem primeiro os objetivos, e aí a gente tem os 6 objetivos aí do programa regional, as metas que compõem esses objetivos, e na primeira coluna aí a gente tem a situação atual, levando em consideração esse exemplo anterior. Então, o país mesmo olha para o que o programa regional está estabelecendo como um mínimo aí, uma atuação padrão nos países, e ele classifica aí como que está a sua situação no cumprimento de cada uma dessas atividades e metas. E a partir daí ele tem uma progressão de para onde ele imagina, para onde ele vê que o país tem capacidade e possibilidade de evoluir. Isso também não é uma obrigatoriedade para o país que ele alcance aí as metas implementadas depois de 5 anos, mesmo porque isso não é algo que o país vai ter que fazer obrigatoriamente, não é algo que vai ser cobrado e tudo mais, mas esse é um ponto de referência para o próprio país identificar as priorizações das ações que ele tem que ter, quais as próximas metas e atividades que ele pode focar para ir evoluindo, progredindo para a implementação de todas as metas previstas no programa regional. Então, o próprio país avalia a sua condição atual, ele avalia a sua situação atual, para os objetivos e metas estabelecidos e dentro de suas possibilidades, qual a perspectiva de avanço que ele vê para o seu país no período de 5 anos. E levando em consideração apenas essa primeira coluna aí da situação atual do país, a gente chega, então, nesse tipo de gráfico que a gente chama de gráfico de radar. Então, a gente tem aí cada um é como se fosse um relógio, né? Cada um das pontas aí dos 12 itens são cada uma das metas, e a gente tem aí classificação em vermelhinho para as metas não implementadas, em amarelo as parcialmente implementadas e em verde as implementadas. E aí a gente consegue identificar as principais fortalezas e dificuldades que o país exemplo tem, né? E qual vai ser o papel do Panaftosa nesse processo todo? Então, primeiro, realizar o segmento aos países. Então, seguir junto aos países os avanços alcançados e a situação atual das atividades, metas e objetivos, ou não, em seus programas. Prover cooperação técnica a partir disso, levando em consideração as atividades, metas, levando em consideração a matriz de evolução do país, a gente consegue identificar, junto ao país, quais as prioridades e para onde ele vai nos próximos anos, e por meio da cooperação técnica, prover aos países, de acordo com a necessidade única de cada um, priorizando as ações e contribuindo para os avanços, Então, eu coloquei aqui 3 exemplos recentes que nós estamos trabalhando. Então, é um projeto para implementação de programa mais ou menos nessa estrutura no Peru, revisão da norma técnica para o tema com o Senazica, que é a área de sanidade de defesa animal do México, capacitação para vigilância na Guiana. E um terceiro papel aí, uma terceira função do Panaftosa dentro do programa regional é ser um mediador aí regional para obtenção de insumos, materiais e tecnologias, levando em consideração que tem países que não detêm dentro do seu mercado acesso a todos os itens necessários para o estabelecimento de um programa de controle de raiva em herbívoros, mediar a comunicação para a realização de ações conjuntas em fronteiras. A gente também teve um exemplo recente que foi um foco grande que ocorreu na fronteira entre Equador e Peru, e aí trabalhar na mediação de ações conjuntas do lado do Equador, do lado do Peru, para que as ações de controle fossem implementadas igualmente, né, e estandarizadas nos 2 países, assim como distribuir informação dos países aos países das Américas, identificando os avanços e ações aplicáveis de um país para o outro. Quais as nossas perspectivas com o programa, né, para o futuro? Primeiro é ter uma informação e dados precisos e representativos sobre a nossa região, né? Segundo, os países, se vocês, como o Brasil, são exemplo disso, executa uma quantidade enorme de ações, executa o programa da forma como ele é estabelecido, mas essas informações, elas muitas vezes elas não são procuradas, não são dados os devidos holofotes, a devida atenção para o mundo de todo esse trabalho que é realizado por parte da defesa animal do Brasil. E isso acontece também para os outros países. A partir do momento em que você tem os dados representativos de toda a região, de todo o trabalho executado, você tem uma visão geral de tudo que a região, o continente americano como um todo, vem trabalhando no controle da raiva e os diferenciais dela. Então, agregar informações ambientais, movimentação de animais, de comportamento, direcionando melhor as ações, identificando melhor os perfis epidemiológicos que a gente tem no nosso continente, e avançar em novas práticas, ações e tecnologias com constante atualização do programa regional, de acordo com os novos conhecimentos gerados, né? E com isso avançar a região como um todo, para você ter a enfermidade controlada de forma conjunta pelos países. Então, aqui eu apresento as referências ao trabalho, e eu gostaria, então, de finalizar agradecendo e deixando aí o meu e-mail de contato. Obrigado.
Obrigado, Felipe, pela sua palestra. Nós recebemos algumas perguntas pelo chat, e eu queria aproveitar esse tempinho que ainda nos resta para a gente discutir, tá? Eu vou fazer pela ordem para a gente não se perder aqui também, né? A primeira é uma pergunta e também um pensamento, né, uma ideia que foi deixada aqui, né? Diz o seguinte, né, o serviço veterinário oficial, central, o Ministério da Agricultura, no nosso caso, não poderia, junto ao Ministério da Saúde, fomentar campanhas de mídia massiva na TV e no rádio para a conscientização dos produtores no combate à enfermidade? Essa foi a primeira pergunta. Um comentário.
Muito obrigado, professor. Sim, na verdade, a própria Ellen e o pessoal do Ministério da Agricultura, que está aqui presente na reunião, a gente já vem realizando também, e a gente vem participando aí como ouvinte, mas de reuniões aí de integração entre Ministério da Agricultura e Ministério da Saúde. A gente caminha cada vez mais para esses sistemas de trabalho integrado, ainda mais no âmbito de One Health, quando a gente pensa, né, de uma saúde, ainda mais em zoonoses e raiva, que é um problema central, um problema muito característico para o trabalho da agricultura, da saúde e do meio ambiente em conjunto. Então, ações de integração, discussões de como fazer a integração Entre os eixos já é realizada ações nesse princípio, como, por exemplo, quando você tem foco, e você tem aí a comunicação por parte da saúde pública, mas também ações futuras aí de como integrar melhor os dois sistemas, né, tanto de sanidade animal quanto de saúde pública. E não há exemplo do Brasil, outros países nas Américas também já realizam esse tipo de integração. Então, o Brasil ele vem realizando sim, esse tipo de trabalho, foi um comentário bastante pertinente. O Brasil tem sido exemplo nesse ponto, sim.
Obrigado, Felipe. Tem uma outra pergunta que diz o seguinte, é mais curtinha. Além de dar suporte aos países, existe alguma expectativa de atuação regional do Panaftosa?
Sim, como a gente falou, a gente trabalha no princípio da cooperação técnica. Cooperação técnica, o próprio nome já diz, é cooperar tecnicamente junto com o país na execução, na realização de atividades e aplicação, exportação desse conhecimento gerado para outros países, né? Então, o papel do Panaftosa é realizar, sim, essa coordenação de ações regionais, identificando os conhecimentos e ações aplicáveis de um país para o outro e promovendo esse avanço na questão da execução das ações dentro do âmbito de um programa regional agora que a gente está estabelecendo.
Certo, Felipe. Tem mais algumas aqui, tá? A terceira pergunta está perguntando se é possível estabelecer a porcentagem de casos diretamente relacionadas aos morcegos. Como avaliar a origem dos morcegos e se existem propostas de prevenção da raiva nos morcegos? Eu acredito que no âmbito das discussões, as perguntas devem ter sido feitas no âmbito das discussões das legislações, enfim, né, da implementação dos programas, né?
Sim, sim. Na parte da legislação, o animal predominante, quem trabalha na parte de defesa animal, vai poder confirmar essa informação. No Brasil, e a exemplo dos outros países das Américas, o desmodos rotundos é o 99,99% dos casos, ele é, sim, o responsável pela transmissão aos animais domésticos. A gente tem exceções, tem casos específicos em um país ou outro, como, por exemplo, acho que Trinidad e Tobago, que tem casos também de raiva canina sendo transmitida aos animais domésticos de produção, mas predominantemente, não sei dizer em porcentagem assim exato, mas uma porcentagem grande, esmagadora, ela é transmitida por desmodos rotundos. Existem várias formas de você identificar isso, a principal dela é por meio das agressões, e por isso que a gente defende que as agressões têm que ser, sim, um ponto importante de vigilância antes da ocorrência dos casos, né?
Uhum.
Essa é uma das formas de você avaliar aí a origem dos casos, mas nem sempre a agressão ela é perceptível pelo produtor e também pelo agente público de defesa animal. Mas você não pode perceber num animal específico, mas se você tem no rebanho alguns casos de agressão e outros que você não identifica agressão, mas você tem a sintomatologia, você pode fazer aí o vínculo epidemiológico dentre esses animais. E existe uma proposta de prevenção da raiva nos morcegos. Essa é uma questão bastante discutível, bastante polêmica, existe pesquisa em cima disso, mas ela ainda está no âmbito da pesquisa, ela ainda está no âmbito de ideias. O que a gente tem de concreto no momento de ferramenta que a gente tem agora para utilizar é o controle populacional. Existem vários pontos para serem discutidos, evidenciados e pesquisados sobre a prevenção da raiva nos morcegos, mas isso, como estou dizendo, precisa de mais pesquisa, precisa responder várias questões ainda para a gente chegar no entendimento de que essa seria uma ação. Mas no momento que a gente tem de ferramenta para controle da raiva é o controle populacional de uma espécie silvestre que cresce artificialmente, se apoiando nas alterações que o homem vem promovendo nos últimos séculos, não é agora, é uma alteração que vem desde a chegada dos europeus na América e a vinda do animal doméstico para cá, né? E a criação da pecuária intensiva pela nossa região.
Uhum. Felipe, acabou de aparecer uma pergunta Está relacionado a isso que você falou, eu vou adiantar essa daqui, tá? Dizendo o seguinte, essa vigilância, eu imagino que é essa que você comentou, né? Seria as capturas e o controle dos abrigos? Falam que não é tão recomendado devido à migração de colônias. Tem algum comentário a respeito disso?
Tenho. A vigilância não é a realização do controle. A vigilância é você identificar as agressões, é você identificar a ocorrência dos abrigos de desmodos rotundos e também a vigilância de casos suspeitos. Aí, quando a gente entra na parte de controle da espécie transmissora, do desmodos rotundos, aí a gente está entrando em ação de prevenção, né? Existem os estudos, sim, que dizem que a prevenção, que a adoção de ações de controle populacional, elas podem auxiliar aí na dispersão da raiva, mas também a gente vê que quando ele é realizado de forma eficaz, inteligente, direcionado, e você realiza o controle total ali do abrigo, você consegue, sim, impedir essa transmissão, porque a população de desmodos rotundos, por meio do controle, ela fica muito baixa nessas áreas de intensa produção animal, e aí você não tem densidade animal de morcego suficiente para um transmitir para o outro, né? Então, você realiza o controle da população. E então, por isso, você vai... Agora, como eu apresentei no slide, quando você não realiza um controle eficaz, você não realiza um controle total, você não tem identificação de todos os abrigos cadastrados, não realiza monitoramento desses abrigos para verificar se a sua ação de controle foi efetiva ou não, você não está realizando de forma correta a medida de controle populacional e você vai ter aí, sim, um desequilíbrio gerado pela ação, mas se a ação for realizada de forma inteligente, eficaz e total na área onde você vai trabalhar, ela sim traz grandes benefícios no controle da raiva. Inclusive, a gente pode perguntar aí para os próprios agentes de sanidade animal dos estados e também do nível nacional e dos níveis municipais que estão aqui presentes na reunião, eles mesmos vão dizer que nas áreas onde eles têm bastante conhecimento sobre os abrigos, eles realizam o controle nesses abrigos que eles conhecem, eles conseguem acabar com a transmissão da raiva ou manter por longos períodos de tempo com uma transmissão muito baixa ou sem transmissão de spillover para os bovinos. É justamente nas áreas onde tem maior dificuldade de controle populacional é onde você tem uma maior ocorrência de casos de raiva em herbívoros.
Ok, Felipe. Complementando essa pergunta, tem algumas perguntas que foram feitas, especialmente pelos veterinários dos serviços oficiais. Depois eu faço um comentário final, só para a gente não perder esse encadeamento, tá? Mas tem uma outra pergunta aqui, né? Se na prevenção de agressões aos animais, quais seriam as ações indicadas aos abrigos e morcegos? Você já comentou, né, alguma coisa a respeito disso, mas eu não sei se tem alguma complementação que você acha pertinente.
As ações de prevenção, quando a gente olha para elas, a gente vai ter as ferramentas que a gente dispõe hoje, são o controle populacional. Por que o controle populacional? Porque existem dois fatores aí na ocorrência da enfermidade. O primeiro é você ter a enfermidade ocorrendo entre os transmissores, e a segunda causa da ocorrência da enfermidade é você ter animais suscetíveis a receber a doença. Então, a ideia do controle complementar à vacinação, né, a recomendação de vacinação para os animais que estão sendo agredidos, você impede. Você realiza uma ação tanto a nível de circulação do vírus entre os animais que são responsáveis por esse spillover para os animais domésticos, como você protege os animais domésticos de eventuais ocorrências da enfermidade no morcego para eles. Então, você precisa das duas ações, uma que é para diminuição do risco e da circulação do vírus na população, que mantém ele no meio, que são os desmodos rotundos, e também a vacinação, que vai proteger no caso de agressões.
Uhum. Tem uma pergunta, vou encadear uma pergunta com isso que você acabou de mencionar. A pergunta diz o seguinte: existe uma porcentagem do que é mais eficiente, a vacinação, eu imagino que seja essa pergunta, ou o controle populacional dos morcegos? Talvez
essa é uma
esforço,
pergunta.
né? Vacinal ou esforço de controle, né?
Sim, sim. Qual dos dois é? Olha, você pode realizar a vacinação pela vida toda. Você pode jogar essa responsabilidade para o setor privado, e o setor privado vai vacinar pela vida toda, e isso nunca vai impedir a ocorrência e a circulação da raiva. E a exemplo do que a gente vê também na raiva canina, que a gente pode, aí, de forma bem grosseira, tá? De forma bem grosseira, fazer uma tração paralela entre o animal doméstico de produção e o homem, que também é um elo final aí na transmissão da raiva canina. Você pode ficar estabelecendo a vacinação do homem pós-exposição para todo sempre, mas o vírus vai continuar circulando na população canina, o vírus vai continuar circulando na população de morcegos desmodos rotundos. Então, não existe uma que é mais eficiente que a outra. Elas duas são complementares e são unidas, porque você protege tanto com a circulação do vírus entre os animais responsáveis pela manutenção dele, como você protege os animais que podem aí, eventualmente, receber o vírus, se tornarem infectados por conta desse spillover entre os que mantêm e os que são suscetíveis, né? Então, não tem como você traçar essa é melhor do que aquela, elas são complementares e não tem uma que é mais importante que a outra.
Uhum. E para complementar ainda isso que você mencionou, sobre especificamente os morcegos, a pergunta diz o seguinte: a subnotificação das agressões de morcegos é um grande problema no meio rural, sobretudo em regiões mais distantes dos grandes centros. Quais medidas você entende como efetivas para corrigir essa deficiência?
Essa é uma pergunta muito boa, né?
Sim.
Porque você tem diferentes manejos, diferentes produções animais na nossa região. Você vai ter os pequenos produtores, você tem aí as produções também de leite, onde o trabalhador rural está em contato constante com esse animal, e você também tem os casos aí de produções extensivas, de longos territórios, de largos territórios, onde você não tem contato constante e frequente com os animais, né? Então, aí a gente tem diferentes aspectos, né? A questão da agressão, primeira coisa é que o produtor ele tem que ter a consciência de que, se ele notifica a agressão, identifica e notifica a agressão, porque quando eu falo de notificar a agressão, eu não estou falando unicamente desses casos de produção extensiva, eu estou falando de todos os produtos, inclusive aqueles pequenos e intensivos que têm contato direto com o animal, mas não fazem também notificação de agressão. Muitas vezes porque eles não entendem os benefícios que a notificação dessa agressão vai ter, não entendem muitas vezes como que a defesa animal vai atuar frente a essa agressão no apoio a ele, os benefícios que isso vai trazer para a produção dele. E você também tem países, como eu mostrei, que realizam quarentena, isolamento dos prédios, que você não pode movimentar animais quando você tem casos positivos. Então, isso também para o produtor é algo, ao medo da sanção, também traz para ele aí essa questão de não fazer a notificação. Então, esse é um trabalho para você conseguir conscientizar o produtor de que ele precisa notificar a agressão quando identificada, é um trabalho que você vai ter que construir com a confiança que ele vai desenvolvendo aí com a execução das ações junto com a defesa de sanidade animal. Nesses casos onde você tem aí as produções muito extensivas, o que acontece muitas vezes é que esses animais são agredidos longe do contato do produtor, do trabalhador, esses animais eventualmente acabam morrendo, e quando o cara tem contato com os animais, ele vê que está faltando alguns animais, e as causas da morte aí, ele não vem a identificar exatamente quais que são, né? Então, você tem que verificar muito bem o tipo de produção que a gente está falando, e a questão de você ter o produtor junto com você para identificar e apoiar a defesa animal na identificação das ocorrências de agressão, que isso vai trazer benefícios para ele, uma vez que isso vai desencadear ações por parte da defesa animal.
Felipe, eu vou fazer uma última pergunta minha agora para você. As demais perguntas, muitas delas, e eu acredito que a maioria das pessoas que fizeram essas perguntas vão participar de um outro evento que a gente vai fazer com os veterinários do serviço oficial, que começa amanhã, esse evento inclusive. Então, essas perguntas serão abordadas com bastante profundidade em 2 dias, especificamente para isso. Então, a gente vai guardem essas perguntas para amanhã e quinta-feira. Mas uma última pergunta minha, a gente tem 2 minutos, tá? Só, antes de terminar o tempo. Mas a interlocução do Panaftosa com os governos nacionais é louvável e muito positiva, gerando aperfeiçoamento de políticas públicas e ações de campo. Como é a interlocução do Panaftosa com os pesquisadores destes países? Seria interessante que eventos ou oficinas entre pesquisadores fossem organizadas pelo Panaftosa?
Boa pergunta, professor. A construção dos programas, o nosso principal ator aí, a gente existe para atender o país, né? Tudo que a gente faz é dos países para os países. Então, quando a gente estabelece o desenvolvimento e a construção de um programa regional, por exemplo, como esse que eu apresentei, ele é porque os países demandam disso, os países constroem isso, e o nosso trabalho é dos países, né? A questão do envolvimento da academia é de suma importância. O principal papel e fundamental aí da academia é gerar a questão, né? É questionar as práticas que são realizadas, é questionar o conhecimento que existe e procurar as respostas para isso. E a gente se embasa muito no conhecimento gerado pela academia. A gente tem, por exemplo, a gente tem aí um contato estreito com várias universidades e pesquisadores, com você mesmo, professor Ricardo, e de várias outras universidades das Américas, no desenvolvimento aí de conhecimentos e tecnologias. Seria bastante interessante, sim, o desenvolvimento desses seminários, desses eventos de discussão do tema, mas eles acontecem dentro de um contexto específico, né, para desencadear respostas e entendimentos das necessidades dos países. Então, havendo a necessidade dos países, a gente faz, sim, o desenvolvimento desses questionamentos, e a gente considera bastante, sim, o conhecimento gerado pela academia no desenvolvimento do nosso trabalho. Quando a gente coloca as ações propostas, é baseada no conhecimento gerado pela academia para poder embasar o que a gente vai recomendar aos países, somado também ao conhecimento empírico do trabalho que as defesas de animal realizam, tanto da parte de defesa animal, parte da agricultura, quanto da parte de saúde pública nos ministérios de saúde, também pelo conhecimento gerado de forma empírica pelo trabalho dessas pessoas, né? Então, tudo é somado, e a academia tem suma importância, sim, no estabelecimento das recomendações que a gente vai passar para os países.
Felipe, eu queria finalizar agradecendo a sua participação. Foi excelente a sua palestra. Acredito que todos tenham gostado. Pela quantidade de perguntas, eu acho que ainda tem bastante tema para ser discutido, quem sabe num outro evento, né? Mas obrigado de coração pela sua participação, tá bom? Se você quiser
eu que agradeço
conosco,
mais uma vez, professor. Obrigado. Obrigado a todos.
Postos. Hello, Daniel. How are you? Daniel, I'll introduce you in Portuguese, all right? How are you?
There seems to be a little delay. I think I can hear you.
A little bit, but it's okay.
Yeah.
I'll introduce you in Portuguese, right?
Sure, that's fine.
Bom, muito obrigado, Daniel, novamente pela sua participação, por ter aceito o nosso convite. Daniel é um pesquisador sênior da Wellcome Trust, baseado no Instituto de Biodiversidade, Saúde Animal e Medicina Comparativa e no Centro de Pesquisa de Vírus da Universidade de Glasgow, no Reino Unido. Ele recebeu seu diploma de graduação na Universidade da Virgínia, em 2004, e seu doutorado em Ecologia da Universidade de Geórgia, em 2011. A pesquisa do Daniel combina estudos de campo longitudinais de morcegos selvagens, filogenética, estudos comparativos e modelagem epidemiológica para compreender, prever e prevenir a transmissão de doenças infecciosas entre as espécies. Seu trabalho na América Latina nos últimos 15 anos tem se concentrado em como os conhecimentos ecológicos e evolutivos podem guiar um controle mais eficaz da raiva transmitida por morcegos hematófagos. Ele publicou mais de 65 artigos revisados por pares, Recebeu vários prêmios, incluindo o Prêmio de Ciência, o Sci Life Lab para jovens cientistas, o Prêmio Philip Leverhulme e o Prêmio Wellcome Bait, e é explorador emergente da National Geographic. Daniel, muito obrigado novamente por ter aceito o nosso convite. O tempo é seu. Obrigado.
Okay. Thank you, Professor Ricardo, for the invitation to present in the symposium today, and also to the other presenters who have been presenting their work and will present their work over the rest of the week. It looks like a very informative and fascinating lineup of presentations. I'd also like to thank all the participants, the people from Brazil and other countries around the world for tuning in and listening to hear all about the work that's going on both in research and in management. So in my presentation, I'm going to focus on how myself and my research team have used kind of insights and techniques from ecology and evolution to produce insights that we think have some management relevance for vampire bat populations and for vampire bat rabies in particular. So this is the bat that we're all focused on, the common vampire bat, and you've already seen this map before of its distribution from northern Mexico in the north to Chile, Argentina, and Uruguay in the south. As we also already heard, the reason why this bat is such an important transmitter of rabies is that, like many other bats, it maintains a transmission cycle of bat-to-bat transmission as the bats bite each other, and when they get the infection, they die. But this bat is extremely important and different because it is exclusively blood feeding. And so when these bats are feeding on humans or domestic animals, it presents the opportunity for cross-species transmission. The previous talk also really nicely gave an outline of the economic and the public health burden of vampire bat transmitted rabies. So fortunately, that talk had more detail than I had available, but I think we're overall aligning in the perception of the levels of risk. So across Latin America, thousands of cases of rabies are happening in livestock every year attributed to vampire bats. And in many countries across Latin America, vampire bats are the primary cause of human rabies. We have currently several options for how to prevent the transmission of rabies to humans and domestic animals. The first of those is that we could potentially break the transmission cycle of the virus in the wild bat population. And so, as that's already been discussed, the principal way of doing that is through population control using a transferable poisonous paste called vampiricide. The way that the vampiricide works is that it's smeared on one bat or several bats, which is released. The other bats will then groom the first bat, which was treated. When they do that, they ingest the poison and they die. And so that has clear benefits in terms of reducing the bat population and in terms of reducing bat bites on humans, domestic animals. But what we're really interested in is how effective it is for rabies prevention and how it might be improved. The second area where we might be able to control this disease better is through preventing rabies cases being transmitted to humans and livestock. And so again, as we've heard, the principal way of doing that is through vaccination. Our vaccines work at the individual level, meaning it protects a human, it protects a cow. The limitation is that, at least in terms of human rabies vaccination, it's often implemented reactively. So after people already begin dying, a vaccination campaign begins, which is not ideal. There's a similar problem when it comes to livestock vaccination. Preventative vaccination should be done at a wide scale, but it is costly, and in countries where the stakeholders, like the local farmers, need to be paying those costs, that prevents widespread adoption of livestock vaccination. And so that results in a significant burden of rabies cases, despite the fact that some animals are being vaccinated. There's also reason to think that there is room for improvement in all of our strategies for rabies control. This is a map showing the increase or decrease of rabies in livestock in all countries throughout the Americas where vampire bats occur. The ones in red, we've seen increases in the burden of rabies through time. The ones in blue, some decreases in the burden of rabies through time. And I should note that in some of these cases also, we've seen expansions geographically in the areas of the country where rabies is a problem. So we've certainly seen that in Peru, potentially in Uruguay, potentially in Mexico, where the virus is affecting livestock and people in geographic areas where historically, maybe even just 10 years ago, the virus wasn't present. So what we're trying to do research-wise is try to understand how, if we know more about the ecology of rabies, the ecology of vampire bats, what options does that open up in terms of improving current management and in terms of guiding the development of new management tools? So in this presentation, I'm going to focus on 3 areas of my work where I think we've shown findings that have either direct or indirect implications for the prevention and control of rabies. The first of those is thinking about the spatiotemporal dynamics of rabies. And particularly, I'll show you how we've combined evidence from field studies in wild bats, from mathematical modeling, and from viral phylogenetics to show that rabies is locally epizootic, but regionally endemic. And I'll explain later on what I mean by that. The second part of the talk will focus on this topic that's already gained a lot of interest in this symposium, which is culling vampire bats. And so when I say culling, I'm talking about population control using the vampiricide poison. And so I'm going to ask, what do we know about the efficacy of that practice for preventing rabies transmission, both within bats and from bats to other animals? And in the third part of the talk, I'll be presenting some much more recent work exploring the possibilities that vaccines might have for controlling rabies within wild bat populations. So can we vaccinate bats to prevent rabies transmission to humans or domestic animals? So I will start with the spatiotemporal dynamics. The main question we want to ask here is, why doesn't rabies go extinct from vampire bat populations? It's actually kind of a curious question because this is a lethal virus which kills its host, and the bats reproduce very slowly, only 1 offspring per female per year. And so you would think that either rabies would wipe the bats extinct or the virus itself would go extinct and the bats would be fine. But that doesn't happen, obviously. So there's a couple of different hypotheses for why rabies doesn't go extinct, how the virus maintains itself over long time periods. The first of those is something that I'll call the local persistence hypothesis. What you're seeing in the diagrams here are kind of these circles representing individual bat colonies, and the lines represent a connection between those colonies. So that's bat dispersal. And so under this local persistence hypothesis, you have the amount of rabies varying through time. So initially a small amount of rabies, and then more, and then less again. But the crucial thing is that rabies is always present in these bat populations. We can contrast that with the alternative hypothesis, which I'll call metapopulation persistence. In this case, we have the virus starting out in one area, spreading to a different area, and then at the later time point, spreading to a third and fourth area. But crucially, the places that had rabies in the very beginning and in the middle have now lost rabies. So what is going on here is that at the colony level, the virus is invading and then later going extinct. And so distinguishing those two hypotheses actually has some implications for how we might think about rabies prevention and control. If the local persistence hypothesis is correct, then we might be able to control rabies by keeping bat populations small. But on the other hand, if rabies is always there at some level, that means we'll need to be constantly vaccinating livestock, just because there will always be some risk of transmission. In contrast, under the metapopulation hypothesis, it could alternatively be possible to control rabies by stopping the spread between colonies. And similarly, if we're thinking about prevention of cases in livestock, if we can anticipate when and where the virus is going to spread, then we might be able to vaccinate high-risk areas before the virus arrives, effectively preventing those deaths from happening. So we've been trying to investigate this problem for almost 15 years now. And the first way that we started doing it was by starting up a longitudinal monitoring study of vampire bats. At the moment, we're following 43 colonies, which are spread across the coast, Andes, and Amazon of Peru. In all of these colonies, we catch bats using traditional methods like mist nets and harp traps, and we give all the bats an individually numbered wing band. An important thing that we do when we catch these bats also is to collect a blood sample. And with that blood sample, we're able to use a virus neutralization test to determine whether or not the bat had antibodies or did not have antibodies. If the bat did not have antibodies, then we're assuming that it hasn't been exposed to rabies recently. But if the bat does have antibodies, then we'll assume that the bat was exposed probably within the last 6 months or so to the virus. And so using this kind of combination of longitudinal monitoring and antibody testing, we can monitor individual bat colonies through time and quantify the proportion of individuals within each colony that was seropositive. And so what you're seeing here is just the data from the first 3 or 4 years of that study, where the white parts are the proportion of the colony that's seropositive, so the ones that have antibodies. And so in this study, that varied between about 10%, the average was 10%, and that varied between about 5% and 30%. So we can use that sort of time series information to compare different mathematical models of rabies transmission. And it's not very important for the purposes of this talk what the different mathematical models are, but I just wanted to make a couple of points which arose from this mathematical modeling exercise. Essentially, what we're trying to do is create models which reproduce the antibody data that we saw within the wild bat populations. The first finding that we saw was that no matter what biological assumption we make about the pathobiology of rabies within vampire bats, the estimated R0 value is always below 1. And so probably everyone is familiar with R0 now in the context of the pandemic. But in this case, R0 is just the number of secondary infections that occur when a new rabid bat is introduced into a fully susceptible bat colony. And so because the R0 value is estimated to be less than 1, that suggests that the disease is going to fade out from a single bat colony. And that implies that the rabies virus cannot persist within a single bat colony. You need multiple bat colonies interacting with each other in order for the virus to persist. The second piece of evidence that I wanted to show was this one, where we were looking at the parameter space, which created the best reconstruction of the patterns of serological data that we saw in the wild vampire bats. What you're seeing here is a likelihood surface where higher values basically mean closer correspondence of the model prediction to the field data. So these dark areas here are kind of the best, the places where the model was performing best. And we looked at that across a variety of values of 2 parameters. The one that's really important for the purposes of this talk is this immigration term. And so we're quantifying here how much immigration of rabid bats do you need into a focal colony in order to explain the patterns of that we saw in the wild bats. And so what we saw was that actually you need a relatively high amount of immigration to match the patterns observed in the field. This is just a selection of the simulations from this region of parameter space. And the thing really to look at here is just the black line, which is the seroprevalence. And so just as we saw in the wild bats, this simulated seroprevalence is varying between about 5% and maybe up to 25, 30%. And again, the average is 10%, just as we saw in the field data. So it does a reasonably good job at reproducing what we saw in the wild bats. And importantly, we can look at other parts of parameter space and see that that does not happen. So if we were to reduce the amount of immigration, which again, you can think of as like the dispersal of bats between colonies, we see extremely low and sporadic seroprevalence. So not at all like what we saw within the actual data. So these 2 pieces of evidence together are strongly arguing against local persistence. So meaning that a single bat colony cannot maintain rabies indefinitely. But there's a limitation to all of this, which is that serology, looking at antibodies, is a really indirect way of studying viral circulation. And we only had a couple of years of data. So we needed ways to kind of better explore this possibility of maybe rabies is able to persist endemically within a small area. So with that in mind, I was approached a couple of years ago by Bernal Leon, who's from the Ministry of Agriculture in Costa Rica. And he was discussing with me the problem of vampire bat transmitted rabies in that country. And to me, Costa Rica stood out as particularly interesting because it's a small country, but it's one where rabies has been reported almost every year since the mid-1980s. So this is just data from 1986 until 2014. And so that combination of being a small country with apparently endemic rabies provided an interesting opportunity to investigate whether it really was kind of staying within Costa Rica permanently, and some of the year-to-year variation is driven by bat demography or environmental variation, Or is what we're seeing this recurrent extinction recolonization dynamics? So are we seeing viruses invade into Costa Rica and then disappear? So with Bernal, we carried out a phylogeographic analysis of vampire bat rabies viruses, which were collected from livestock in Costa Rica between 2004 and 2017. This is the main phylogenetic tree describing those results. And there's a lot to take in here, but I'll try to just talk you through the main findings. First is that we saw 5 genetically distinct viruses. So these are the ones that are where the branches are colored in gray. All the other cases where the branches are different colors, those are rabies viruses from other countries, but all vampire bat rabies viruses. So we see 5 different groups of vampire bat rabies which had circulated in Costa Rica during the time period of that study. But what was actually really interesting about this was that most of those viruses had common ancestors with viruses from other countries. So in blue, these are viruses from Colombia. Again, down here, viruses from Colombia. And this clade of viruses from Costa Rica is also related to other viruses from Mexico. And so that suggested that there was certainly some role for international movement of the virus and explaining the patterns that we saw within Costa Rica. The samples from Costa Rica also had information on the date and the location of sampling. And so that allows us to look at kind of how long each virus was persisting within the country. And we could see that in general, viruses weren't sticking around for very long. For example, you can see this red virus, which was only found in 2006 and 2007, and never in other years. And the only single virus which was present at the very end of the study in 2017 had only been circulating in the country since 2011. And so through some somewhat complicated statistical and phylogenetic analysis, we came to an estimate that on average, these viruses were circulating in Costa Rica for just over 10 years and then disappearing. The next thing we were able to do was to fit some statistical models to these patterns of virus presence and absence. And so I did that using something called a multinomial regression. And what you're looking at here is the output of that model, where you're looking through time, and on the x-axis and the y-axis is the probability that that strain of rabies is present. And so it stood out fairly clearly that we had some viruses like this virus 1C and 1A, which were absent from Costa Rica at the beginning of the study and appeared later on, whereas other viruses like virus 2 and virus 1D were clearly present at the beginning, but then disappeared longer into the time series. And so we think that this shows that although rabies appeared to be persistent or endemic at the national level, what we're actually seeing is a serial pattern of virus extinction and reinvasion. So that's at the country level very consistent with this metapopulation persistence hypothesis. But we still needed to really explain where all these viruses were coming from, the ones that were apparently invading into Costa Rica. And so we did another Bayesian phylogeographic analysis where we were able to quantify rates of virus dispersal in between countries. And so the really fascinating thing that we found from doing that was we found statistical evidence of viruses invading into Costa Rica from the south and then spreading north, and also patterns of southbound viral spread. So viruses invading from Mexico into Costa Rica and then spreading from Costa Rica down into Colombia. And so it seems like Central America is forming a corridor for rabies viruses to spread between North and South America. And that is kind of epidemiologically interesting, but it also provides some potentially new options for prevention and control of rabies. One of those is that we might be able to use this information to strategically target bat populations through things like use of population control to potentially prevent the virus from invading into new geographic areas. And that might be particularly effective if we combine population control with knowledge of the natural geographic barriers to viral spread. So whether that's a mountain range or a water body, whatever it happens to be. Also, from a livestock vaccination perspective, it means that potentially if we can understand how the virus is spreading between regions, between countries, we might be able to vaccinate animals before rabies has arrived. Finally, I think this study really highlights the critical importance for international data sharing and international viral sequencing. Without the data that we had from other countries like Mexico, Colombia, El Salvador, we would have never been able to reconstruct what was going on in terms of viral movement between countries, which is, it seems, is quite fundamental to how the virus persists in this region. So that's just my brief call for the real need for improving genomic surveillance for vampire bat rabies. Okay, so I think from that first part of the talk, we've seen fairly clearly that the virus is not persisting locally within single bat colonies, but instead needs to move around between colonies in order to persist over long time periods. Next, I'm going to talk about culling vampire bats. So the conventional wisdom is that fewer bats should mean less rabies. We can make that a little bit more kind of mathematically explicit by thinking about what the relationship might be between the density of bats and the relative amount of rabies transmission. And this relationship isn't really well understood, but could have a number of different forms. In the absolute best case scenario, we're in this red line, where you reduce the bat density to a certain threshold, and the virus is no longer able to persist and goes extinct. We wanted to test whether we saw any sort of relationship between bat density and seroprevalence, and so between bat density and rabies transmission. But again, the best data that we had available at the time was seroprevalence. So again, this is looking at the proportion of bats in a colony which have rabies virus neutralizing antibodies. And we did that using the first 4 years of data from Peru, and we found no significant relationship between colony size and seroprevalence, which suggests that if we were to artificially reduce bat colony sizes, we might not expect to see a great reduction in the amount of rabies transmission. The more controversial and worrying result from this study, though, was that when we looked at bat colonies that were never culled versus ones that were periodically culled or regularly culled, we saw actually higher seroprevalence in the bat colonies that were periodically culled compared to the ones that were never culled. And so that is a bit worrying because it suggests that culling might either be ineffective, so not reducing rabies transmission, or counterproductive, actually increasing rabies transmission. But there are a huge number of caveats with that study. And I think I just want to point those out here. One of those is that this was an observational study. We weren't experimentally culling these bats. And so it's entirely possible that the areas that had more rabies already might have been culled more, rather than culling causing more rabies death, more rabies activity. Second is that since this wasn't an experiment, we only know about the levels of culling and the type of culling that was undertaken by the Peruvian authorities at the time of our study. And so it's entirely possible that more culling or different culling could have been more effective. The second limitation is, again, that we were only measuring antibody prevalence, whereas what probably is more interesting and more important is the number of rabid bats in an area, and consequently the amount of rabies transmission to humans or domestic animals. So what we really wanted to know was, does culling vampire bats actually reduce the burden of rabies in livestock? Many years passed before we actually had the opportunity to test that. And that came about through a program run by the regional government of Apurímac, and again in Peru, where this program was about a 2-year initiative with about a budget of about $1 million US. And it was titled Capacity Strengthening for the Prevention and Control of Vampire Bat Rabies in 25 districts of the provinces of Abancay, Andahuaylas, and Chincheros in Apurímac region. So to give you a little bit of details of what that program involved and where it was, this is a map showing you the areas in red, which are the districts which were part of this program. And so these are the ones where both bats were culled using the vampiricide paste, and things like livestock vaccination was supported. A key thing to point out from this map also is all of these orange dots. And so all those orange dots are rabies cases that were reported between 2013 and I think 2019 in this map. And so you can see that there's a tremendous amount of rabies activity in this area. Indeed, it's about 100 to 250 cases that get reported every year, even though there's probably many more than that happening, but just don't get reported because of sub-notifications. So that intensity of rabies transmission was important because it provided statistical power to be able to test whether the culling activities actually reduce the amount of rabies transmission to livestock. So in terms of the bat control itself, the vampiricide poison was applied to over 21,000 bats across many different districts of the region. It's important to remember that this is a spreadable poison, so 21,000 is really the absolute minimum number of bats that were killed. Probably many more than that were killed because the poison does spread. We were also able to get data on livestock vaccination, and that was really important because if rabies declines, that could be because of culling, or it could be because there was an increase in livestock vaccination. So we needed to control for that. In addition to culling and vaccination, the authorities in this area also conducted questionnaire surveys on bat bites in livestock. And that was really important because it gave us an idea of the bat population size. We looked at this in two ways. The first was in the percentage of animals on any given farm that were bitten. And that reduced fairly substantially from around 25% at the beginning of the project to around 10% at the end of the project. So that shows that the bat population size almost certainly decreased. Yet when we looked at the percentage of farms with at least one bat bite, we saw that did not decrease very substantially. So it went from maybe 75% to something like 60%. And so taking these two graphs together, it means that the bat population was smaller, but most of the locations where there were vampire bats before the population control campaign, most of those places still had some vampire bats after the population control campaign, but probably just fewer of them. But the important thing and the really interesting thing that we wanted to study was whether the population control of bats actually reduced rabies transmission to livestock. And so to ask that, we looked at this time series. So this is actually two time series of the number of rabies cases in livestock from 2004 to 2019. And I've separated this out into districts that were culled, so part of the population control program, and those that were not part of the population control program. And this gray area is actually when the culling took place. And so straight away, we can see one obvious finding, which is that after the culling campaign ended, we still had plenty of rabies cases happening in livestock. And so that implies that culling certainly didn't eliminate rabies from the bats. But the actual curious finding here was that maybe there was a bit of a decline here, but it seemed to be happening both in the culled districts and the districts that weren't even part of the population control program. And so that could happen for two reasons. It could be that the areas that were culled are really important for determining the dynamics or the amount of rabies in areas that were not culled. Or it could just be that what we're observing here is a natural fluctuation in the amount of rabies transmission, and that natural fluctuation is happening both in the culled and in the non-culled areas. So we couldn't really distinguish those two possibilities just by looking at the data. So instead, we fit something called Bayesian state space models. This is essentially a model which describes the spatiotemporal dynamics of rabies along with extra variables to describe the vaccination and culling efforts. So this figure that I'll show you next is going to give you the outputs of that model, where you'll see posterior distributions of effect sizes on each model parameter. And so anytime these effect sizes overlap with 0, it means that that factor wasn't necessary to explain the observed dynamics of rabies. So I'll talk you through the main effects. First, we saw strong evidence of spatial effects. All this really means is that if there was an outbreak in a neighboring district, either in the same time period or in the time period before, then that heightened the risk of a rabies outbreak in a focal district. And that makes a lot of sense because we know that rabies can move around on the landscape. So having rabies nearby is a risk factor. Second, we looked at vaccination of livestock. And these effects were initially quite counterintuitive because we were seeing positive effects of vaccination on the number of rabies cases in livestock. And so that makes it seem almost as if vaccination could be increasing rabies transmission, which obviously doesn't make any biological sense. However, it's important to note that vaccination was measured in two ways. One was 12 months before, and one was 12 months after any focal time point. And the much stronger effect was the vaccination after a given time point. And so what we think we're seeing here is simply that animals are being vaccinated more after rabies cases begin to be detected. And we think that there's sort of some spillover of that effect into this other measure of vaccination before any focal time point. Finally, we'll get to the culling effects. And here, in both cases, whether we were looking at culling going on within a neighboring district or in the focal district, we saw that the effects were sitting right on 0. So at least in this study in Peru, we're not able to find any effect of population control of vampire bats on the incidence of rabies in livestock. Even though it didn't seem to reduce the incidence of rabies, it's possible that culling might have slowed the spatial spread of the virus. And so one thing that I didn't mention at the beginning of this section was that in addition to having all of the data on rabies incidence and culling efforts, we were also able to obtain brains from many of the cattle that died of rabies. And so from those brains, we obtained 300 either complete or near complete viral genome sequences. And these were samples that were collected between 1997 and 2016. So before and during the culling period. And this bar chart just shows the number of viruses that we sequenced, and that almost all of them were from the same virus lineage, which we're calling lineage 3, although we had some viruses of another genetic background, lineage 1, which were detected in the later years. I'm just going to focus on the lineage 3 today. So how do we actually estimate whether the population control efforts increased or decreased the rates of viral spread? The way we approached that problem was through Bayesian phylogeography. Some of you may have seen reconstructions like this, where you can take a virus phylogeny, which has information on the sampling date and the location of each sample, and you can reconstruct how the virus spread across the landscape. However, what we can actually do is reproject that reconstruction of the spatiotemporal spread into a series of dispersal events, where for each dispersal event, we know where the virus started and where it ended up, and we know the distance that it traveled, and we know how long it took to travel that distance. And so together, that gives us an idea of the velocity that the virus had spread. And so we can look branch by branch in the phylogeny and assess whether variables like the amount of population control influenced the speed that the virus was spreading at that place and at that time. So to talk you through how that actually worked in practice, because it is somewhat of a complicated analysis, basically the pipeline was this, where we would sample a single tree from the posterior distribution of a Bayesian analysis. And that was important because Bayesian phylogenetic analyses don't rely on just a single topology. You have many thousands of trees which can be sampled from and are equally likely. So we sample a tree, we use established methods to reconstruct the spatial spread and to calculate the velocities that the virus was traveling at every branch within that tree. And then we test what was correlated with the velocity using generalized linear models. And then in this case, we used an automatic backwards model selection to work out which variables were important to explain the patterns of spread. And so, as I said, we did that from 1,000 different trees pulled from a Bayesian phylogenetic analysis. And that was really important to control for the fact that there is uncertainty in phylogenetic reconstruction. And so each time we did that 1,000 iterations, we recorded the proportion of trees where each variable was retained by model selection. And we recorded the effect size of variables, conditional on them being retained in the model. So these are the results showing the really preliminary outcome of that work. And I emphasize this is preliminary because I've just done it earlier this week. So if there are suggestions on how to improve this, I'm very, very, very happy to hear them. This bar chart shows the proportion of models or the proportion of trees where each variable was retained. And the red indicates the variable being retained and statistically significant, whereas the gray is retained but not statistically significant. And so you can see that the variable which was included in the largest number of models was this sampling intensity. And so that was a variable which was measuring the average number of rabies cases around a given location, and which makes sense because we thought that the more an area is sampled, the more viruses are going to be sequenced, and the more likely we are to detect rare long-distance dispersal events. I won't go through all the other variables just for the sake of time, and I'll focus on the variable where we tracked the effects of culling on virus dispersal. And here we saw that that variable was included in a relatively small fraction of models. So only 12.5% of trees retained that culling effect and had it as statistically significant. And only about 28% of models retain the culling effect at all. So really, it's a small minority of models where we see culling as an explanatory variable for the patterns of virus spread. Nevertheless, we can look at the effect sizes when culling was retained. And interestingly, we saw that it was negative. So this would imply that culling might actually be slowing the spatial spread of rabies. But I should emphasize again that this effect was only observed in a minority of model iterations. So I think we must view this as very tentative evidence that culling might slow the spread of rabies through geographic areas. So to summarize what we can conclude from this study about existing interventions for reducing the burden of rabies, reduction of vampire bat populations is clearly beneficial for reducing bat bites on livestock, but we have doubts over whether it's breaking the transmission cycle within bats or significantly reducing mortality in livestock, though there is still an interesting question emerging from this, suggesting that culling may slow the spatial spread of the virus. In terms of livestock vaccination, we know that it protects individual animals. The limitation with it is that it's often reactive to outbreaks or to perceived risk in the local area. And because it requires outbreaks to be initiated, it is going to be an indefinite need. Also because it doesn't control the amount of rabies within the bat population. So vaccination by itself, as has already been discussed, is not necessarily going to be a sufficient management option because it will have to be deployed indefinitely at effectively infinite cost. So in the final part of the presentation, I want to talk about some very early work that we're doing, thinking about an alternative for managing rabies, particularly not so much the vampire bat population size, but rabies in wild vampire bat populations using vaccines. So as we know from all other cases of rabies in terrestrial carnivores, vaccination of the primary reservoir is a cornerstone for preventing rabies transmission to humans and domestic animals. And so that's done in a number of creative ways, whether that's using vaccine-laden baits or community-based vaccination programs. But the advantage there is that both of those methods allow you to vaccinate a large number of animals. Whereas with vampire bats, we really don't have a great method to distribute vaccines to a sufficiently large number of animals to actually have an epidemiological impact. A potential solution for that is the idea of using transferable vaccines. And so these would be vaccines which are orally effective, but applied in the same way that the vampiricide poison is applied. So applied topically and then ingested by other animals. Yet instead of dying, those animals would then become vaccinated against rabies. This is not a new idea. The idea of using these sorts of vaccines for vampire bats has been around at least since the mid-1990s. And we already have at least 2 vaccine candidates which seem to work effectively, at least in captive bats. The main question is, do these vaccines transfer sufficiently among wild bats to suppress rabies? So we didn't want to test that exactly by releasing vaccines into wild vampire bats just yet. So instead, we've been trying to study how well these vaccines might spread using fluorescent biomarkers. We used 2 biomarkers. One of those is a UV powder, which spreads by bat-to-bat contact, and the other one is rhodamine B. Rhodamine B is particularly useful in the context of monitoring vaccine uptake because it can be applied topically in a gel, just as the vaccine would be. And when it's ingested, it turns the hair follicles of the bat this nice fluorescent orange. And so in doing that, we can deploy this vaccine topically to wild bats and then monitor the rest of the colony just by plucking hair samples over the following weeks and months, and then analyze those hair samples under a microscope to determine whether they ingested this mock vaccine off of the first bat that was treated. And so using some statistical modeling, we found that in the 3 colonies of vampire bats that we studied, each bat was spreading the mock vaccine to between 1.5 and 2.2 other individuals. We then applied those field estimates to some mathematical models. And the idea behind these mathematical models was to estimate would the given observed amount of vaccine transfer actually reduce the amount of rabies transmission? And how would that compare to the same amount of transfer, except using the vampiricide poison rather than a vaccine? And so again, this is a purely a simulation-based project, but I'm going to show the results from like the main results of this work. What you're seeing here is on the x-axis is the deployment effort. So this is the percent of bats that whatever was applied to, whether it's the vaccine or the vampiricide. And on the y-axis, you're seeing the difference in the reduction of rabies cases. And so anytime these lines are going above 0, it means that vaccination reduced the size of rabies outbreaks better than culling. And so you can see that at any relatively low deployment effort, which is the more operationally reasonable, vaccination is performing better than culling in reducing the size of rabies outbreaks. And we saw similar results for the frequency and the duration of outbreaks. So this was all fairly encouraging that these sorts of transferable vaccines targeting wild bats could soon alleviate the burden of human and livestock rabies. Of course, that doesn't completely solve the problem because we still need to worry about bat populations and bat population growth, but it's at least a potential tool on the horizon. The limitation, of course, is that these sorts of vaccines are going to require continuous deployment because they're deliberately engineered not to spread very well. They only spread from the initial bat that was treated to other individuals. And that's going to become expensive. A potential solution to that is to scale up using something that I'll refer to as a transmissible vaccine. And so this is a vaccine that spreads infectiously, kind of in the same way that a natural virus would. And the advantage here is that as this vaccine spreads, it's immunizing more and more bats. And so that reduces deployment needs and consequently reduces deployment costs, increases coverage, and could potentially spread to populations where the vaccines were never released. And that's really just taking advantage of the natural dispersal of the bats to spread the vaccine to new areas. The technological barriers to generating transmissible vaccines are actually much lower than you might expect. The top way to do this is by engineering a naturally occurring harmless virus, which I'll call a vector, to express a single protein from a target virus, which here is rabies. And so you have this hybrid virus, which is mostly a harmless virus, but also contains the surface protein of rabies, which is what would cause a bat to become immunized by it. If we were to take that approach, we need to think about a lot of factors related to safety and efficacy. We want that the naturally occurring vector to have a high prevalence, because that implies that the vaccine might also obtain relatively high coverage in the bat population. We want it to be host specific, and that's largely to avoid any unanticipated effects on non-target species. We don't want a vaccine that could potentially affect livestock or humans in some unexpected way. Finally, and this is where a lot of viruses don't meet the criteria necessary to be an adequate vector in this type of application, is that the virus needs to be capable of superinfection. And so that means that an individual which is already infected with the naturally occurring virus should be able to be infected with the vaccine version. In other words, you can't have immunity from the natural virus blocking the spread of your vaccine. So one group of viruses which has been widely discussed as a potential vaccine vector are the beta herpes viruses. And so many of these, including most notably cytomegalovirus and other primate infecting beta herpes viruses, have already been used as virally vectored vaccines for pathogens like SIV, tuberculosis, and Ebola. But we didn't know if bats, and particularly vampire bats, had a similar version of a beta herpes virus. So we collected samples from bats, fecal samples and saliva samples, and we subjected those to metagenomic sequencing. And using that, we discovered a virus which we've since called Desmodus rotundus beta herpes virus. And so a current PhD student in my lab, Megan, has been investigating whether that vampire bat beta herpes virus meets these prerequisites for being an effective transmissible vaccine vector. So first, she explored prevalence using PCR and found that almost all bats are infected. It's really difficult to find a bat which isn't already infected by this virus. So we think we certainly tick that box. In terms of host specificity, we carried out a phylogenetic analysis of the vampire bat beta herpes virus, along with a number of other beta herpes viruses, which we discovered in sympatric bats from Peru and other countries in South America. And we found that most of these form bat species-specific transmission cycles. And the vampire bat virus was no exception to that, except for one case of the vampire bat beta herpes virus being found in Artevius literatus fruit bat. And so we think that these things, this beta herpes virus may not be purely species specific, but is unlikely to spread beyond vampire bats and very closely related species. Certainly would be unlikely to infect cattle or humans. Finally, we tested superinfection by carrying out deep sequencing of individual samples from vampire bats. We did this just initially with 2 individuals, and what we found in both of them was a mixed frequency of nucleotides at given sites. And so what you're seeing here is just a snippet of the beta herpes virus genome, which is in total about 230,000 base pairs. So you're just looking at a very short stretch, but this pattern was repeated throughout the genome, where we see kind of an equal frequency of 2 alternative nucleotides at the same position. And so that's implying that there's at least 2 viruses present within a single bat. And we've subsequently followed this up looking at individual bats through time. And so we can see that they're actually, as a bat gets older, it's acquiring more and more strains of this virus. So we feel that we are confident that superinfections are possible. A final thing that we wanted to look at was whether this beta herpes virus vectored vaccine would be likely to spread to inaccessible bat populations. So that really depends a lot on the behavior of the bats and the dispersal properties of the bats. If so, that would be really beneficial because it would mean that the vaccine might spread to areas where we never even released it in the first place. So really having a massive boost in the efficacy for rabies control. We analyzed that by looking at the distribution of not just the beta herpes virus generally, but in individual strains of the beta herpes virus in these bat populations. So in total, what Megan has found is that there are at least 11 strains circulating in Peru, and all of them have a somewhat distinct geographic distribution. So we see some which are found exclusively in the southern parts of the country. And I'll just point out here that all the white points are areas that were sampled but did not have that strain circulating. So we see some that are disproportionately in the south, some that are disproportionately in the central and northern parts of the country, and some which are much more widespread. So kind of already infecting all the different colonies of Peru where we investigated. So we think that at the very least, a released vaccine strain will be spreading regionally and perhaps much further. So we think this is an exciting potential advance towards the elimination of vampire bat transmitted rabies. And one of the reasons why we're so encouraged by these results is some recent work by Scott Nussmer and James Bull, who suggested that anytime your transmissible vaccine spreads at a higher rate than the pathogen that you're trying to control, then the pathogen is predicted to be eliminated. So just to talk you through this, what you're looking at is in the red, and kind of the red to orange scale is the reduction in effort required to eliminate the pathogen, in this case, rabies, relative to a conventional vaccine. And so the darker colors indicate more effort. So if you have a highly transmissible pathogen, but a not very transmissible vaccine, then you'll require a lot of effort to get rid of the virus. Where we are with rabies, though, is somewhere over here. We know that the R0 value of rabies is probably just right around 1, maybe potentially less than 1 in some areas. So all we need is a mildly transmissible vaccine to potentially locally eliminate rabies. So obviously, we don't actually know yet the R0 value for the vampire bat beta herpes virus, so we can't say exactly where it fits on this spectrum. But given the high prevalence that we've observed in the field and the frequency of multi-strain infections, we think that it's probably going to be higher than what the R0 value for rabies. Another thing that we don't yet know is the frequency at which this herpes virus undergoes latency and reactivations. And that's going to be important for 2 reasons. First, because latency and reactivation provides an opportunity for continued spread of the vaccine. Every time the vaccine reactivates in a bat, that's a new source of infection for other bats by the vaccine. The second benefit is that at the individual bat level, a reactivation could potentially make this into a self-boosting vaccine because the reactivation will again expose the bat to the rabies virus antigens. So a lot of that is unknown, and I think will ultimately require experiments to be done in captive bats. But I think there's at least the encouraging ecological data suggesting that this could work. Obviously, we're really far away from actually doing any of this, and the next important step that we need to do is to actually isolate the beta herpes virus and engineer a vaccine. So I'll stop there and just quickly summarize what I've talked about. First, I discussed how we've used all this serological, epidemiological, and genetic data to show that rabies is maintained over long time periods by recurrent extinction and recolonization dynamics at the colony level. And so for control, that means we should probably either be synchronizing control over large areas or understand better how the virus spreads to have spatially, temporally targeted control. In terms of vampire bat population control, we've shown that it has benefits for reducing bat bites, but doesn't seem to have very strong effects on the amount of rabies transmission to livestock, although potentially weak effects on the viral spatial spread. In the final part, I discussed vaccines, their potential for use in rabies control, although obviously a great amount of work still needs to be done there. So I'll stop after thanking the members of my research team who did a lot of this work, the funders, collaborators in Peru, the US, and here in the UK, as well as the institutions that have supported this work through providing authorizations and data. So thank you very much for listening, and I'll be happy to take questions.
Thank you, Daniel. We have a few questions, but very interesting ones. I'll make the first one now. Actually, is Paulo's question in mine as well. Sorry, I'll do it in português. Qual seria a consequência da eliminação do vírus da raiva na população do Desmodos rotundos? Essa é a parte do Paulo. E a minha com a vacinação de morcegos, a população não explodiria, aumentando as mordeduras de herbívoros?
Yeah, that's a great question. And to date, there's not really much evidence that rabies is responsible for controlling the size of the vampire bat population. That makes sense intuitively because we know that rabies only kills a relatively small fraction of bats that get exposed to the virus. So some of our earlier modeling work suggested that only 10% of bats that get exposed actually die of the disease. So we think that rabies is a relatively small mortality factor, so that if you were to eliminate rabies, you wouldn't see at least you wouldn't see disproportionately large increases in the size of the vampire bat population. That doesn't mean that you would have a vaccination-only strategy. I think we will always need some way to manage the size of vampire bat populations. As we've discussed, culling may or may not be the right way to do that. I think there's potential for much more exciting and epidemiologically complementary ways of doing bat population control, but thinking about it on the other end of the spectrum. So thinking about preventing reproduction of the bats, maybe using hormonal reproductive suppressants rather than killing the adult bats. And that could be epidemiologically a better choice, because that way you're preventing the birth of susceptible bats, which could get and spread rabies, rather than killing older bats using like vampiricide, which may have immunity from previous exposures to the virus. So I think there's kind of multiple reasons for thinking about alternative ways for managing the bat population size. Some of those are to do with animal welfare, some are to do with the kind of epidemiological efficacy.
Ligando a esse comentário que você acabou de fazer, você acha, é uma pergunta minha, que o controle da raiva em colônias poderia ser feito pela captura e eutanásia de alguns indivíduos, até que um limite, um threshold de endemismo seja atingido?
So I kind of missed the question, but selective culling. Yeah, so I think there is potential for using selective culling if we have a better understanding of when and where the virus will appear. I think the limitation with reactive culling is that we're predominantly culling individuals after the virus has already passed through that colony. Mm-hmm. If you could anticipate which colony will be the next one to get rabies, then culling, at least, I mean, our models even predict would be beneficial for reducing the likelihood that rabies invades into that colony. Although our models also say that vaccination would be a better choice. Of course, vaccines aren't even available yet. So I think for the moment, selective culling, if used strategically, could be a nice complement.
Two more questions. We still have 2 minutes. A raiva já foi encontrada em outros tipos de morcegos, como insetívoros e frugívoros. A interação dos desmodos com estes morcegos não poderia ser também uma fonte de renovação do vírus nas populações de morcegos? Did you get it?
Yes, so that's exactly right. Rabies is maintained in multiple bat species in the Americas. But the curious thing about the disease is that it's actually maintained in largely species-specific transmission cycles. So there is the vampire bat rabies virus, and then there's an insectivorous bat rabies virus, and these things are maintained completely independently. So if you were to eliminate rabies from vampire bats, maybe eventually you could have the virus jumping in to vampire bats from an insectivorous bat or a fruit bat, but this will take time, because we already know that most of those cross-species transmission events do not result in sustained transmission within the novel host species. So there's a lot of good phylogenetic and epidemiological evidence suggesting that that's the case, and it really works to our advantage if thinking about the long-term sustainability of vaccination as a rabies elimination strategy.
Uma última pergunta, Daniel. O que impede, então, o uso de vacinas para os morcegos no campo, já que os resultados são tão promissores?
Okay, so in livestock, if the question is in livestock, the reason why vaccines aren't applied preventatively is really just, I can only speak from my experience in Peru, and there it's predominantly the cost. People don't want to spend the money to get their animals vaccinated unless they're very certain that there is a real threat and that there's an imminent threat. So we've seen that if there's rabies circulating in a community, people are much more willing to vaccinate their animals. But if they haven't had a rabies case in the last couple of years in any of their animals or in the neighbors' animals, then they're unlikely to vaccinate. So it's really just a question of, are they willing to invest in a potential threat? In the case of human rabies, prevention is I think, predominantly limited by the difficult access of populations which are at highest risk. Again, my experience is mostly in Peru, where these are often communities which are only accessible by boat, and health authorities just don't have the funding and the capacity to be able to travel to these areas routinely to vaccinate people preventatively. So often it just means that vaccination campaigns are only taking place after deaths are detected in those populations, sadly.
Daniel, tem último comentário de uma participante falando sobre raiva humana. Eu queria um breve comentário, nós já estouramos o tempo. Você acha que não é estranho que outros focos de raiva em humanos não tenham acontecido após um foco na reserva de Unini em 2016? Ou isso deve ter sido devido à subnotificação de casos em áreas remotas? Parcialmente já foi respondido por você, né?
So the question is, why were there no human rabies outbreaks in recent years? In Peru, there's been actually a major effort to do preventative vaccination. That was a direct result of a large rabies outbreak. In other countries, I'm not quite sure what may be going on. Like, some of this is due to the sporadic nature of the disease. Some of it may be due to underreporting. I think it's very unlikely that if just a few individuals die, especially in a remote community, that it may not get reported. So I think there's a combination of the sporadic nature of the disease and underreporting, which maybe makes it difficult to detect when these cases are happening. It's easy to detect a large, unusual outbreak. It's much more difficult to detect sporadic cases.
Ok. Daniel, muito obrigado pela sua participação. Thank you very much. We appreciate it.
Thanks everyone for listening.
Obrigado. Mas vamos seguir então o nosso evento. Tchau, Daniel. Vamos seguir então o nosso evento com mais uma palestra, a última palestra do dia. É uma palestra. É uma palestra que vai ser dividida por dois palestrantes diferentes, que eu vou apresentar agora. São dois colegas, na verdade, trabalham conosco. O primeiro deles é o professor Gustavo Machado. eu vou fazer uma introdução tanto do Gustavo quanto do professor Joaquim Prada. O Gustavo é graduado em 2010 pela Universidade Federal de Santa Maria, no Brasil, completou o mestrado em epidemiologia veterinária na URGS, na Universidade Federal do Rio Grande do Sul. Ups. Tudo bom, Gustavo?
Tudo bem?
E em epidemiologia da diarreia viral bovina, em 2013. O Dr. Gustavo também desenvolveu ferramentas de aprendizado de máquina aplicadas à epidemiologia, enquanto fazia o seu doutorado em 2016, quando ocupou também um cargo de professor adjunto do Departamento de Medicina Veterinária Preventiva na URGS. Posteriormente, ele concluiu tem um cargo de professor, associado de pós-doutoramento no Departamento de Medicina Veterinária, na Universidade de Minnesota, e em janeiro de 2018 foi nomeado professor assistente no Departamento de Saúde da População e Patobiologia da Faculdade de Medicina Veterinária da Universidade Estadual da Carolina do Norte, para liderar pesquisas sobre epidemiologia transfronteiriça. O principal foco da pesquisa do Dr. Gustavo é investigar a ocorrência e a disseminação de doenças infecciosas, especialmente doenças transfronteiriças, fornecendo bases com base científica para a tomada de decisão no que diz respeito à prevenção e controle. Atualmente, foca em modelagem matemática e estatística tradicional para mapeamento prático da propagação de doenças infecciosas entre populações de suínos. É autor e coautor de mais de 183 publicações revisadas por pares no campo de doenças animais e epidemiologia. Além disso, no laboratório do Dr. Gustavo, há um interesse de longa data no ensino da epidemiologia de doenças e desenvolvimento e implementação e capacitação para serviços veterinários oficiais, nacionais e internacionais. E o professor Joaquim Prada, vou apresentá-lo já também, os dois vão dividir horário. Professor Joaquim é professor de epidemiologia veterinária na Faculdade de Saúde e Ciências Médicas da Universidade de Surrey. Seu foco da pesquisa nos últimos 10 anos tem sido o desenvolvimento de modelos matemáticos e estatísticos para doenças infecciosas em uma ampla gama de patógenos, tanto em humanos quanto em animais. Sua maior ênfase está em informar a tomada de decisões políticas para o controle e eliminação de doenças. Como membro do consórcio de modelagem de doenças tropicais e negligenciadas, ele liderou recentemente trabalho sobre o impacto da Covid-19 no cumprimento de metas de 2030 da Organização Mundial da Saúde. Ele lidera também um grupo de pesquisa na Universidade de Surrey, no Reino Unido, que desenvolve modelos matemáticos para ajudar a controlar doenças zoonóticas, incluindo o lissavírus transmitido por morcegos. Bom dia, Joaquim. Bom dia, Gustavo. Como vão?
Bom dia, bom dia.
Bom dia. O tempo é de vocês, obrigado pela presença.
Tá bem, obrigado, Ricardo, aí, pelo convite. Por questão, de repente, de interpretação, vou partir para o inglês para comunicar com o Joaquim também, e aí a gente vai trocando, pode ser? So, first of all, thank you very much for the invitation, and today we're just going to talk to you about a work that we've been doing since 2019. We got some funding from a partnership between USP, University of Surrey, and North Carolina states, and then we got together with Ricardo and Joaquin to try to develop a model that could be applied for the state of São Paulo as far as trying to understand the spillover from bat rabies from the roost to the cattle population. So I'm going to talk to you briefly about the background, then Joaquin is going to give you the overview of the modeling and then the preliminary results that we have so far. So, of course, this is the work of a team that got together in 2019, and we keep working. So, in my site, I'd like to acknowledge the work of Manuel Jara, which is postdoc in my lab. In the side of Joaquin, we have a bunch of other researchers as well working on this model, especially Eva, that developed most of the modeling part, and also Ricardo, that helped us with the brainstorming, helping, developing the model, and did most of the work within São Paulo and Brazil that helped us actually branching out what we wanted to do, try to understand the spread and spillover of bat rabies in São Paulo, and then try to help the local authorities to build and enhance their surveillance system in the region. So we're going to talk a little bit about what has been done as far as trying to model bats, and then also the modeling done in cattle on infection per se. But really, we want to focus on the model that we built, that is simulation based on some observation, but mostly simulation assumptions, where we take in consideration the reservoir, which are the bats, and we're going to call recipient the cattle, and we build explicitly the environments into this model. And we took a different approach here, where we actually reconstruct the suitability conditions for bat rabies within the region, and also we take in consideration previous work done by Ricardo in São Paulo as far as the dynamics of the roost and dynamics on feeding habits of the reservoirs, which are the bats. And then we build on top of it interventions that are part of the current plan for Brazil, and try to shed some lights there, and try to figure out really what would work best in this case for São Paulo, but also with the idea that it could be applied different states in Brazil, and then maybe be uptake by the regional regulations. Then we're going to discuss with you the preliminary results that we have so far. So Stryker talked a lot about rabies, and he's been doing a lot of work in this area. We all know that, but I just want to bring back a topic here as far as epidemics go, and we all know that there is some doubts of where it's coming from, COVID is coming from. So there is this common sense that about 60% of the emerging diseases are coming between wildlife or zoonotic diseases jumping between different species. Still, we are very behind understanding the process. So trying to shed some lights in a region that we do have good data in the population would be a good approach. That's why we're gonna toggle today. And we still don't know what to do and how to do as far as early detection and trying to guide the activities at the field in order to anticipate a new spillover or a new emerging of something that might be spreading the wildlife. So studying bat rabies can give that opportunity to understand a system that has been studied for many years now, as far as trying to anticipate a new spillover, a new emerging of a pathogen. And just coming back a bit to what maybe you guys talked early in this conversation, but the habits of bat rabies of these models, it can vary a lot. It can go to whatever you have feed available. So the chances of actually bites in human populations that are near to a very high infected region, there is a possibility that could have the cases, could have the outbreaks, but also if there is something else circulating in those populations, we will expect something to actually spill over to the human population as well. But here today, we're going to talk about the spillover to cattle population, which is the main topic. We're going to try to shed some lights here on how can we estimate risk areas, or how can we figure out which areas in space are more likely to be exposed to new outbreaks. So in this case, talking about the recipient, which are the cattle populations. There's a lot that is known and unknown around the disturbance of a system that might be stable. And I'm going to talk a little bit about the migration cycle that is a common discussion in bat rabies, where you might have a population that is infected, and ever so often spills or infect different hosts. It might be some dynamic that is not very well understood. The population might decline, and you might end up with a rooster that is really not infected. And the theory is that it could be infected, or this rooster that is infected could be infected by migration of an infected bat coming from a different roost that is nearby. So to try to focus in the effort, and I point out to these plots down here, which is a paper that came out in 19, which is interesting as far as looking at how the disease is actually propagating in the reservoir and how the disease can be actually propagating in the recipient, in this case, the cattle. And what is needed for that spillover event to happen? How much infection we need to have at roost level? Is that driven by the distance between the roost and the host or the recipient population? What is really in between that? That's what we're going to try to answer with this study. And when you guys talk about, putting a lot of effort in trying to have a surveillance system that can actually pick up areas that are more likely to be infected, that's very hard to do without knowing the dynamics between, when a new infection would emerge from a roost and infect the recipient, which cattle. So that's very difficult, very time consuming, very expensive. And there is this other conversations talking about a selective control of the bats, and you guys talked about that as well. So how that would actually impact the time for a spillover event is also a question. So we're also going to tackle that a bit. And also the other question around the cooling, and if cooling would actually on the roost would actually disturb this migration cycle and then either be counterintuitive on not controlling, but actually causing a spillover event to occur. So this work has been done, some preliminary work in Brazil. So Ricardo has been leading a lot of work in this area. And just a highlight on one of the work that look at the feeding habits of a few roosts. And in this plot, what you see here, the diamond is location of roost, and then the size of the cattle is the population size. And the idea here was try to figure out and follow some roosts to see how those colonies were actually feeding on the recipient. And the main conclusion here was that most of the feeding were occurring downstream. So there was an effect on the altitude of bats stopping on farms that are downstream. So that gave us some ideas of how we shouldn't consider these events when we're trying to build a model that we're trying to recover surface of risk. So we included in our model, and I'm going to explain more in detail, we included an effect on the altitude within the whole state of São Paulo to consider that the chances of a transmission or a spillover event would happen more truly downstream in the elevation than upstream in elevation. So there's a lot of discussion that go around that as well, and I'll get in detail. And the other work that has been done also in São Paulo by Ricardo and his team was trying to figure out the network between a roost and between the cattle population and also roost to roost. So trying to estimate what is distance that a bat could actually travel that could either infect a next roost or could also infect then a farm that is within a given distance. So these shed more light on the dynamics. So the inclusion of a network by distance was needed when we were building this model, and the network would be needed to be constructed by the distance roost to roost, but also roost to cattle, and then vice versa. So these two work by Ricardo help us to start designing this modeling work that we're going to present to you today. So we consider information that was gathered within São Paulo, and then I'm going to show you the data we use also from São Paulo to try to approximate this dynamic between roost to roost and roost to cattle transmission. And previous work, and a lot of work has been done trying to describe the distribution of roost, trying to recover the niche or the suitability conditions where you expect to have bats and therefore more transmission. A lot of computation work has been done on that as well. And oftentimes the work is either isolated in the bat dynamic or isolated at the cattle side, where a lot of work has been done as well, trying to describe a seasonality effect, try to see that the perspective of just modeling one either the reservoir or the recipient. And then lately there is some more effort trying to understand both together and trying to see the dynamics between the spillover from the bats to cattle. And that's what we're gonna tackle today. Just coming back a bit on what has been done as far as spillover dynamics, there is more work done, but a classical study is this study that is trying to understand where the threshold between the dynamic in the recipient and the dynamic in the reservoir, where things go wrong or where things go right for a infection to actually jump from the reservoir into the recipient, but not only to try to understand the dynamic, but also try to understand the effects if we do manage the reservoir alone, if we do manage the recipient alone, and if we manage both together. What is the sweet spot there? What should we be looking at as far as trying to break the cycle of the spillover that is observed within bat rabies transmission to cattle? So that's what we are going to tackle in this model. So for this study, what we used was the data collected for São Paulo state as far as locations of the roosts that were collected till 2018. So we have data here of about 4,167 roosts. And we gathered the data from the cattle population in the region with the same year with the population within each farm. And this data is coming from the Ministry of Agriculture, and the data on the roosts are coming from a project that has been following up the roosts for many years now. So that's our population of interest. So what we wanted to understand is how and where the spillover is more likely to happen given those dynamics that I mentioned at the earlier talk. So first of all, what we created from the roost data, we reconstructed a niche of the bats. So what we done here is basically we collected environmental layers or variables that are explained in literature that facilitates the presence of bats in roosts. So the probability of a roost to be suitable for a bat to actually live on it. So you take those layers, you have your presence data, which is basically a roost, yes or no, so locations that are present with roost, and then you get at your output as a suitability of a specific grid cell. So what we done here, we went to a very high scale, and we went to create these suitability maps to a grid of 5 by 5 kilometers. So we could then, with that data, we could explicitly include the suitability conditions when trying to model the spread between roosts, so roost to roost, and also roost to cattle. So with the idea that if you had a favor condition for a roost to be populated, the spread or the transmission or the migration of the bats would be facilitated. So what you see in this map is a continuous value, which goes from 0 to 1, 1 being the most suitable pixel of 5 by 5 kilometers. And this is used not only on the dynamic of the model, but also where we start our infection. So what we are trying to do here is trying to see a worst case scenario where we start an infection where it's very highly suitable for roost to be present, to be populated against a location that is really poorly suitable for bats to actually survive. So we explicitly consider that in the model. And thus, when moving forward, after we created this layer, we construct the model, and Joaquin gonna take over the talk now. I'm gonna advance the slides for you, okay?
Yes, thank you, Gustavo.
Thank you.
And also add my thank yous to Ricardo for inviting us, for inviting me to come here today. It's definitely an honor to be here and see so many great talks on rabies today. And I know there are more coming in a few days. So really exciting. So now just to talk a little bit about the modeling framework. So here on the right-hand side is just a diagram of what the model look like. So here we're taking we're looking at the contact network. So we can imagine that the red circles are our roosts, and then the triangles would be our farms. And so what we say here is that we have our roosts are connected with each other, so have this bidirectional relationship. So a roost may infect any other roost, and obviously that goes both ways. But when we're thinking about the connection between the roosts and the farms, we have a directed connection. So only a roost may infect a farm. Obviously, a farm is not going to spread rabies back to a roost, for example. Right, and as mentioned already by Gustavo, we are assuming here, and this is one of the constraints, and there's a number of constraints that I will mention throughout. Is that we're assuming that indeed that connection from a roost down to a farm is going to go down in the elevation, right? So the roost has to be higher elevation than the farm. In the first instance, we're assuming that the maximum range of the bat is around 10 kilometers. So from each roost, it can spread up to 10 kilometers. But we modulate that transmission intensity based on the distance. And for that, we use a gravity model. So for a gravity model, the context is basically that the further they are away, then obviously the less transmission there is. That is fairly intuitive. But also you account for the size, right? So you would expect that, for example, a big roost will transmit more, or a big farm will also kind of attract more animals potentially for feeding. And as Gustavo also mentioned earlier, we also account for that suitability. So the suitability of the environment will also modulate how transmission, how much transmission actually happens. Next slide, please, Gustavo. So now I'm going to go into a bit more detail of what's happening inside the roost and inside the farms. So we actually heard earlier Dr. Stryker talking a lot about different modeling approaches, and so here is just again a slightly different approach, and here we're taking very much like a population-based approach in terms of the roost. So in this instance, we're not necessarily looking at individual bats, but we're looking at roosts themselves, right? And so we can have a roost that is either susceptible or it can be infected, and we say a roost is infected if there is at least one infected bat. We actually divide the roosts between bachelor and harems. So bachelors are mainly and obviously mainly males, where a harem is basically populated by slightly older females. So some of the constraints that we added when we're looking at the dynamics between different roosts is that there's no transmission between two harems directly. So basically, for two harems to be infected, there should be some kind of male moving in between that maybe visits one harem and then visits the other harem, right? There's also a recovery rate. So in this type of models, we always need to think that after the roost becomes infected, there'll be some kind of clearance at some point that is happening. And again, this is in line with some of the ideas that have been mentioned already earlier today. So we expect that a roost might kind of become fully susceptible eventually over time. And this transmission rate that we have, which are the betas that you see here, this obviously, as I said, it depends on the network, right? So it depends on the connections and this transmission rate parameter. Next slide, please, Gustavo. So now think about how the spillover happens. So now we're kind of zooming out a little bit. Before I was looking at that SI, which is the top bit there. Now think about how that then moves to the farms. So the idea is that an infected roost might infect a farm, a susceptible farm, and that farm will become then what we call exposed. And here is just we're looking at farms that have been infected, but we have not detected the infection yet. So they've been infected for a little bit, but we don't know yet. So we are not doing anything about it. We're not putting any kind of control in place necessarily. And so after a period of time, which is at tau, then that farm basically becomes infected in the sense that we detect now the infection, and therefore we can put in place our control strategy of choice. In this case here, we're assuming that there's a lot of immunity after 1 year since the last vaccination, and this kind of accounts a bit for some of the dynamics in the population. And that's something that I'll talk about, I think, later on in the limitations, because in this kind of framework, we have our roosts are statics in terms of population size. So we're not necessarily modeling this kind of repopulation, say a culling takes place and then a roost location is empty and then new bats will move in and form a new colony, for example. So those kind of dynamics, we're not modeling them here yet. So here we just kind of, in the simplest scenario, we have this static kind of population sizes. So the network is static and the network is not going to change over time unless we do any kind of intervention, which I'll talk about in a second. Thank you, Gustavo. We can go to the next slide. Okay, so then, so first we need to start by calibrating the model. Now, today I don't want to go through the details of this. It's a fairly long process because as we mentioned earlier, the network is quite big. We have over 30,000 farms. We have over 4,000 roosts. So it's a big network. It takes a long time to stabilize, and and it's a kind of arduous process. But we can calibrate the model to get some of these variables that we don't know, to get some values for them. And so in the first instance, what we're trying to do is just a very simple toe exercise, which is what I'll be talking today about, which is to just observe what would be the propagation through the network after a single introduction, right? So we've got the data, as Gustavo mentioned, from 2018, and we've calibrated the model to that so you can understand the transmission dynamics that are happening. Now we're gonna let's backtrack for a second. Let's take this whole network, this big complicated network. Let's put an introduction and see how it propagates. And obviously we can repeat that multiple times. So we want to consider in particular 3 different settings, as Gustavo mentioned. So particularly this high suitable environment. So there's what we know transmission is going to be really that are really good for transmission. And these are like our worst case scenarios, essentially. We can also look at areas where we have they're low suitable, so that's like transmission might be more rare, more difficult, and we can also consider something in between. Next slide, Gustavo. So today I'm gonna only talk about mostly high suitable roost results, introductions, and I'll briefly mention some results that we had for the low suitable settings. Next slide. So the other thing that then we want to consider is, okay, well, now we set this introduction, and now we want to run the model for 1 year. So we're just going to have a very short time period. We put an introduction, we're running for 1 year. The introduction is random. It can be any roost. So it's just one random roost, but it's picked from whether it's the high suitable or the low suitable or the intermediate suitable. And then we explore, okay, what would be the interventions that we could do, and how does that change what the propagation that we observe. Obviously, here all the interventions are in response to disease detection in the farm. So we're not covering some of the other aspects that were mentioned earlier, such as early vaccination and things like that. In this case, we're just looking at kind of the reactive vaccination or potential reactive culling. So we have 4 interventions that we consider. Obviously, we can run a no intervention scenario, which is just a counterfactual to give a bit of context. What would happen if we actually were not doing anything? I assume that everyone here in the room agrees that this is not a sustainable way to approaching this, but it's kind of like it's in a sense, it's just a counterfactual. We will talk about revaccination in farms. So once we have infection confirmed in a farm, we can vaccinate in a ring around it of 10 kilometers. We could also explore roost cooling. So we are only going to cool the roosts, and the roosts are going to be cooled again in a 10-kilometer around the confirmed farm. Or we could consider doing both the ring vaccination and the culling. And again today, next slide please, in the interest of time, and because we actually don't have all of the results yet, because again this is still running in the cluster and results are being generated as we speak. So I'm going to focus mostly on this ring vaccination in farms, and I'm going to tell you a bit about culling in when we add also culling on top of that. Next slide, please, Gustavo. So enough talking about the methods, and let's talk a little bit about the results and what we observed. So for today, I just have all of my results have in the same format, which is this type of histogram. So basically, what it's showing is that we are running multiple repeats of this setting. So we're looking at 1 year after an introduction happens, and in this case, we are vaccinating in this area around each confirmed farm. And so what we're looking at here is the distribution of the number of outbreaks that we see at the end of this year period, 1 year, after a single introduction in a roost. Okay, this is for a high suitable environment, so the best for transmission. And what we see here is that we have fairly broad distribution. It has a long tail. You can see up to 72 farms that are, these are confirmed farms that have outbreaks after a single introduction, and this is in just 1 year, right? Now, this is in, again, the most suitable environment. Now, the average number of farms infected is around 13 in this case. Next slide, please. Now, just to give a bit of context, if we consider, well, how does this change when we're looking at the low suitability, right? So when we are doing an introduction in the roost that is, you know, bit more remote, that has, you know, the worst kind of suitability for spreading rabies. What we see here, that's the overlapping green. So what we see is that the distribution has shifted to the left, which is what we'd expect, right? So now we see slightly less farms infected. The mean is actually a bit lower. It's only 11, so it went down from 13 to 11, so a little bit less. Again, the tail is a little bit shorter, so but you can see that, you know, we still see a lot of infection even in a low suitability transmission. So even when the suitability is low, we can potentially still see a lot of spread of disease. And again, these both are when we're looking at vaccination in response to a confirmed infection in a farm. Next slide, please, Gustavo. Now here, now we changed, and now we're looking at what happens if we add culling, right? So we are back into our high suitable environment situation. The blue columns are still the same ones as before. So this is high suitability. We are vaccinating in response. We have on average around 13 farms infected and up to 72 farms infected in that in a particular repeat that we had. And this is all 1 year after the first introduction. Now, when we look at what happens if we add culling, now in this case, what we are simulating here is that we are doing culling in the area around the infected farm. And what we say is basically say, if we're doing a culling, all of those roosts are essentially gone, so they cannot transmit anymore, right? So those roosts are essentially, you know, they disappear from the network, so they cannot transmit onward. So what we see is that the distribution, again, shifts to the left a little bit, as we expect, but the results are actually fairly similar to when we were looking at this low suitability environment, right? So in the situation where we are in a high suitable environment, and we're doing we have this introduction happening, and we're doing both vaccination and culling. The benefit of adding culling to our vaccination, it's fairly marginal. If we think that if we think about the possible range of suitabilities that we could see, and this again in the context of Sao Paulo region, right? Next slide, please, Gustavo. So now let's look a little bit about what's happening in the roosts. So in the roosts, again, after 1 year of the introduction, we see less roosts than farms infected overall, which was actually quite interesting. Now, it's important to bear in mind that there are many, many, many less roosts in this data than farms. As we said, there's 4,000 plus roosts, and we're talking about 70,000 plus farms, right? So obviously there are much less roosts in the data. So in a sense, we see less roosts infected than we see farms infected. But in a sense, it does make sense. The average that we see is around 8 roosts infected after that first introduction. Now, similar as before, next slide, please. If we think about what's happening in this low, in this low, low suitability, now we get that very few roosts are actually infected. So transmission can be quite slow, and actually we have less than 1 roost on average in the simulations that we run. Again, I should caveat this up from the beginning. This is we are still running simulations of this, so this is a relatively small number of runs, between 500 and 1,000 repeats, and we're still running a bit more simulations. So these are very preliminary results, but just to give a little bit of flavor as to what this is looking like. So this suggests that even though there are not many roosts are getting infected, because there are so many farms compared to roosts, actually you could get a number of farms infected just from this one particular roost, if they're close enough. And Gustavo can go to the next slide, please. So now we now change back to our scenario where we're looking at vaccination against vaccination plus culling, and you can see that the results here are essentially very similar to what they were in the previous figure. So again, culling leads to less roosts infected than when we are obviously only vaccinating cattle, but still the average is maybe around 2 roosts. So we still have results which are fairly similar to what we see in this low suitability context. So this was just to give a little bit of flavor of some of these results. I said it's work in progress. Gustavo, next slide, please. So now I want to highlight some of the limitations of this work, because again, I said, obviously these are preliminary results. So we're still working on this as we speak, and there's a number of simulations that we want to run. The simulations, the model was fitted in a Bayesian framework. We heard a little bit about Bayesian already earlier today. So there is uncertainty in the model. There is stochasticity. There's sort of like these effects that are captured in the model. So obviously there's like a lot of variation, and so we need to make sure that we capture that appropriately and we can report on that appropriately. Hopefully, the figures I was showing here, where we're looking at these histograms, could highlight a little bit what kind of, what is the kind of range of variance that we're observing, at least in some of these preliminary results. The second point to consider is that the biological and disease processes are being kept relatively simple. As I said, the model is quite computationally expensive to run as is. That means that a lot of the biological processes had to be simplified. So we're looking at population level approaches. So we're looking at roosts or farms units as the units. We don't have the populations are also fixed in time, as I mentioned. So the only way they would the network changes is when we apply vaccination or culling. Otherwise, the network is static. Now, obviously, that is not necessarily realistic. We do expect some shifts, and as Gustavo mentioned earlier on in the introduction, and as it was said earlier on also in previous sessions, we do expect this population drifts and population changing in sizes, etc., which obviously would have an effect in the transmission. In the results that I presented here, I'm talking about these very simple theoretical scenarios where we're looking at, we have the network designed, and then we're trying to look at how it propagates through the network. And we're assuming here that we're in a fully susceptible environment. Now, in reality, we have a history of transmission. So we've got a number of roosts that are infected, not just one. We have a number of farms that might have been vaccinated recently. So there's a lot of other considerations that we are capturing the model when we were calibrating it, but obviously to explore some of the underlying, to explore some of the underlying effects of how is transmission happening between the roosts and the farms. We try to just simplify to get the most basic ideas. So one single roost is infected, how does it propagate? Right? I mean, there's no way to get, I guess, simpler than that, essentially. Another aspect to consider is obviously the timelines. So in this case, we're running short timelines. We're running just 1 year. Now, a lot of the durations that we need to consider are a bit longer than that. So, for example, we mentioned that we have this return from vaccination back to susceptible in the farms, and that takes a year, a bit more. We heard also earlier about how long it takes for a roost to become susceptible again. And again, this is a longer time, it's longer than 1 year. So in this case, even though we're capturing those dynamics in the model, because these particular scenarios that I presented here today are quite short, those dynamics don't necessarily have a big effect on our results at the moment, which is fine, because what we want to look at is more the effect that we have on transmission, the effect of the network topology, and the suitability. And obviously, the last point to mention is that we have not talked here about the spatial results and the spatial dispersion per se. So how does it actually look when we put this on a map? How it's actually spreading on a map, right? So I talked about how many roosts are getting infected. I talked about how many farms are getting infected, but I didn't say, well, how far are all of these farms, all of these roosts from that original first roost that got infected, right? And it is something that, again, we do have in our results. We still haven't had the time to analyze all of this yet. This is, again, what we call hot code. So we're still kind of running and looking at a lot of these outputs. Next slide, please. So just now to summarize, as I said, this is ongoing work, but and obviously the simulations were long to run. This is even in the context of one state. This is just running one state. Now we want to expand this to, say, whole of Brazil, or or other countries. You can imagine it can be, it's quite big, it's quite slow. So we need to think about ways that we can make this approach scalable. It's important to consider that it does require a fair amount of information, in particular because we're looking at the network topology. It does require knowing where the farms are, where the roost are. So we have a number of scenarios that we still need to explore, but we have hopefully given today some tidbits of some of the features that we've been observing. Next, so I guess one of the main conclusions is that this preliminary analysis, what it suggests is that when you have appropriate vaccination in place, bad culling has a very limited impact. And I'm quite happy that actually this echoes some of the results that we've seen, independent results that Dr. Stryker presented earlier today. So it's good to see that we are agreeing in broadly speaking of what we see. So even the most advantageous scenario, which in this case is the high suitability, the place that is the best for the transmission. Even in that scenario, once you have this vaccination, ring vaccination, adding culling actually does not have necessarily a huge impact. I mean, it has an impact similar to maybe being in a slightly less suitable environment, right? And just to finalize, it's important to think that this variation in transmission that we see in terms of the variation in stability, it's larger than the effect that we see by carrying out culling on top of the vaccination. So just the variation of the stability itself is bigger than the variation that we see just by adding culling, the reduction that we see just by adding culling to a good vaccination program. And with that, I just want to thank our funders from the Global Partnership Network, who funded this tripartite project, and very happy to take questions at this point. Thank you.
Obrigado, Gustavo. Obrigado, Joaquim. Foi ótima palestra. É bom ver os resultados do trabalho reunidos num único lugar. Muito bom mesmo. Temos algumas perguntas, sim. Eu vou começar de trás para frente. Tem uma pergunta do Daniel Stryker, que acabou de vir aqui. Deixa eu só copiar aqui. Só um minutinho. Primeiro, ele parabeniza a palestra de vocês. Ele quer ver muito o aspecto espacial também. Pode ser uma boa maneira de abordar alguns gaps de conhecimento, pois há bons estimadores do quão rápido a raiva pode se espalhar. É muito bom também considerar o culling parcial, por exemplo, a redução de uma colônia, não a total eliminação, e se isso ainda não foi feito, né? Como não foi mencionado, é um comentário. Na verdade, é um comentário, não é uma pergunta. Obrigado, Daniel, pela pergunta. Vocês querem complementar, falar alguma coisa a respeito disso?
Indeed, the spatial thing is what we want to get as the main goal at the end. So provide the state of São Paulo within some ideas of where things would spread more easily. And then, as Joaquin mentioned, the idea is to do it for São Paulo, but then expand it to the whole country. That's gonna require us to do some, maybe try to change languages here, maybe to do some low-level computer language to make that happen. But the idea is to just shed some light and help the states in Brazil to figure out where they should be focusing their effort and not waste, the time where it's not really appropriate. And the partial culling and the other interventions, we have not. The data is good to do that, so that's something that we might need to consider. Trying to do that. As the time goes to run all the scenarios, take a long time. We just need to be a bit strategic where we're gonna put our resources as far as clusters go. So yeah, good. We agree, and thanks for the comment.
Uma outra pergunta. Além de bovinos, outras fontes de alimentação foram consideradas e incluídas no modelo?
Okay, I can take this one. So so the short answer is no. So what we're looking at here is we're really interested in the transmission between the roost to the cattle farms. So in a sense, we know that they're going there for feeding, right? We don't actually care whether the bats are feeding off some other, if they're going to some other host to feed, because that would not lead to transmission to a transmission event that we necessarily care about, right? So in the context here, we just have we're just looking at the roosts, the farms being visited by nearby bats from nearby roosts, and obviously the reason that they would visit is for feeding, but from the purpose of the model, it doesn't matter why they're going or if they're feeding or not. It's just the fact that they are going, and so they can potentially transmit.
Perfeito, Joaquim. Tem uma outra pergunta interessante. Algumas das perguntas eu fui comentando no próprio chat, mas tem uma muito interessante do Rafael, do nosso colega lá do Instituto Pasteur, falando sobre um aspecto interessante do Artibeus lituratus, ou Artibeus de forma geral, né, desse gênero, morcegos desse gênero, serem reservatórios da mesma linhagem do vírus da raiva isolado em desmodos. Ele pergunta, como vocês veriam este fato como uma variável a ser considerada nas estratégias de eliminação da raiva? Por exemplo, uma colônia de desmodos sem infecção poderia infectar uma colônia? Só para adiantar o que eu falei no chat, né? Uhum. Essa transmissão interespecífica, ela ocorre, de fato. E o morcego desmodos, ele também, ele faz também a briga, né? Se abriga com diferentes espécies. Uhum. Mas o aspecto interessante é saber se essa transmissão é suficiente para manter um nível de endemismo, por exemplo, ou se ela serve para a introdução de novas linhagens somente.
Yeah, I mean, I can just briefly comment on that. I think, indeed, so if you have some other reservoir hosts that say another bat species that can help maintain, so what would happen is that when we calibrate our model, we might be overestimating some of the transmission between the roosts because we are only including the Hematophilus bat roosts, right? So if there are other roosts that are contributing, well, the bat species that might be contributing, what might be happening is that we overestimate a little bit how transmission is happening between the roosts that we do model. From a point of view of how does it change the strategies, I think it basically depends what strategy you're going for. Obviously, if you're doing a strategy that is not specific, so you are vaccinating cattle, then obviously you don't necessarily, it doesn't necessarily matter too much as long as you know which area is at risk, or if you're doing that in response to a confirmation, then it doesn't necessarily affect you that there might be some other bad species in the background. But I think the quite interesting, for example, the discussion that the striker was mentioned at the end of his talk about, say, a vaccine that could spread, and then if our strategy then relies on something like having a vaccine that spreads through the population, then actually having multiple species that interact is very important, and saying that, okay, could this vaccine spread to these other species that where they could be acting as a potential reservoir, or maybe help in a sense, be a reservoir, so those are the things where we need to that those are questions I think that obviously they need to be investigated further, and it's very, I think, a very exciting area of research.
Nós somos suspeitos para falar isso, Joaquim. Obrigado pela resposta. Tem só mais um tempinho, eu tenho duas perguntas. Uma que eu trouxe lá das outras palestras e acho que vale a pena trazer aqui para essa última palestra com o que vocês disseram, né? Que se ao invés da vacinação reativa, será que uma vacinação na forma de campanhas, por exemplo, concentrada em uma determinada época do ano, seria mais eficaz?
Yes, so I think, so indeed, so if, and it was, I think, mentioned also a bit earlier by some other speakers, so if we can predict, which is actually the main purpose of this work eventually, as Gustavo has already mentioned, the main purpose is to find the areas that we know are high risk. So if we can vaccinate in advance these areas that are high risk, we would expect then a much better response, bigger reductions than doing a reactive vaccination, because as we said, by doing reactive vaccination, you have things that have already probably moved on, and you are possibly a bit late to the party in a sense. I don't know if Gustavo, you want to add?
Yeah, I totally agree. The thing is, moving forward, that the project was collecting the roost data is no longer as active as it was. So it will be, a good effort to maybe reactivate that. I don't know how it's funding in Brazil at the moment, but to be able to then have a model that can be run yearly and then generate these areas yearly, and not something that we do look in 2018 as for, we don't know we don't know which strain is actually emerging or spread within the area. We have no idea about that at the point that, relying on something that is from the past can help, can be of a help, but the best scenario would be if we actually had the data as far as population goes for those roosts to then calibrate the model every year and push it for the next year, as it's been done for other species. And food animals is very, very easy to do, but without that information, it will be difficult. But maybe there is some movement within this field that could raise some funds to continue that. In some states that are more problematic, but yeah, trying to focus in some areas. We cannot, you have no conditions to do this in the whole country. And, I come from the south, from Brazil, and try to do that in the whole, 400,000, sites is just not feasible. So I think that's a really good idea. It's just a matter of how good the data is to make that happen. Yeah.
Tinha uma pergunta a respeito disso, porque isso não é feito nos outros estados, né? E tem uma outra sugestão, né? Aí é uma sugestão minha, mas talvez as análises de nicho ecológico possam ajudar a complementar a falta de dados sobre os abrigos. É lógico que ter dados sobre abrigos é melhor, porque a gente pode localizar exatamente onde é que devem ser feitas os manejos, mas análises de nicho ecológico podem ajudar.
It has been used, for so many cases, so
Sim.
it'll be one way to do it, but not the best.
Agora, a última pergunta eu deixei para o final, essa porque ela é um pouco polêmica. Mas a última pergunta diz respeito ao seguinte, é super pertinente e é muito, muito boa a pergunta. Com várias pesquisas feitas no estado de São Paulo, por que a raiva continua em ascendência? Que saia justa, coloquei vocês agora.
I mean, I guess from the mathematical model perspective, what I would say is that, as I said before, we're always behind, we're always chasing. So it's always behind the party, right? So, and I think it was highlighted very nicely by some of the work that Dr. Tiger presented in Peru, for example, that, by the time you're going to a place, you know, it already has moved on essentially. So you're always kind of trying to catch up. And really what we need to think about is a more integrated approach where we can combine the modeling, we can combine that for, you know, a lot of the field work, the genetic information that we have. And I know later on next session, we'll talk also about the economics, so consider also the economic implications of all of this, and really take this integrated approach so that we can try to get ahead for once, because I feel to me that we're always a little bit behind. But that's what I would say as a mathematical modeler, not necessarily rabies expert. Gustavo, maybe.
Maybe Ricardo, what do you think? What's your thought on that?
Eu tenho alguns comentários, sim, né? O Estado de São Paulo, ele é mais estudado porque há uma maior disponibilidade de dados, há mais dados, e isso vai de encontro ao que o Joaquim falou, né? Se a gente está seguindo, estamos correndo atrás do prejuízo, o importante é que haja uma informação de base para que a gente inverta essa lógica, né? E, infelizmente, a quantidade e a qualidade de dados do resto do país ainda é baixa. Então, isso só puxando agora um gancho para um evento, até me perguntaram se pode participar desse outro evento que eu mencionei anteriormente, mas isso vai ser amplamente discutido com os gestores do programa de raiva essa semana, né? Quais são os principais parâmetros para colocar energia que geram uma maior eficácia ou eficiência, efetividade dos programas, né? Como priorizar? E eu acho que é por isso, né? Agora, a quantidade de dados não implica na resposta, né? Então, mesmo que haja produção de muitos dados científicos, a transformação disso numa política pública, primeiro que não depende dos cientistas, né? Mas depende de um envolvimento dos pesquisadores com os gestores públicos. É o que a gente está tentando fazer, né, no Ministério da Agricultura. Então, é polêmico, mas é bom, polêmica, né? Bom, nós não temos mais perguntas, o nosso tempo acabou. Eu queria agradecer muitíssimo a presença de vocês dois. Vocês estão em lugares completamente opostos do mundo. Um está indo dormir, outro acordou agora. Mas é um prazer estar com vocês novamente e poder discutir esses assuntos, tá bom? Eu queria só fazer, antes de finalizar o primeiro dia do nosso evento, só fazer um aviso, né? Na verdade, um agradecimento, né, a esses palestrantes tão seletos que nós tivemos, né? Eu acho que o encadeamento das palestras foi muito bom, né? E a gente teve um aspecto regional, depois um aspecto muito individualizado, muito específico, né, com o que o Daniel trouxe para a gente. E agora nós voltamos a esse aspecto mais geral, né, de programa. E então, eu acho que a mensagem que fica é que a incorporação desses aspectos da biologia, né, tanto dos desmodos, como da transmissão do vírus, são importantes na elaboração das políticas públicas, né? Então, eu acho que essa é a principal mensagem. Então, relembrando a vocês, no dia 30, no último dia do nosso webinar, o nosso moderador será o professor Vitor Gonçalves, que é professor da Universidade de Brasília, ele que vai fazer esse papel que fiz hoje. E eu queria também agradecer o apoio da Mariana Dornelas, que também faz parte desse projeto conosco, e que tem dado um grande apoio e tem produzido resultados interessantíssimos, tá bom? Então, eu finalizo daqui. Muito obrigado pela presença de vocês. Espero vocês na sexta-feira, então. Até mais.